Hi, good afternoon, everyone.
Hello.
Hello.
Good afternoon.
Good afternoon, everyone.
Good afternoon. Good afternoon.
Hi, Professor.
Good afternoon, Dr. Sandar.
Yeah.
Hello, Subramanya ji. Namaskar.
Namaskar. Namaskar.
I'm seeing Subramanya after a long time. Ravi
Yeah,
here.
I know. It's quite long time.
Yeah.
Yeah, I think you are here from 2000.
2000 he left.
Yeah,
He left?
No, 2002 or 3 you left, right?
99 he came, 2002.
After that, I came for one conference, maybe around 2008 or something like that.
Yeah.
CTDD.
On the behalf of CDRI family, unfortunately, it's a webinar platform. We would have loved to meet you in person, and we would have loved to interact with you and know the exciting work that you're doing. And for all the students and staff who are joining us online, as well as we have YouTube viewers who are watching this webinar on YouTube platform. As we all know that AI and machine learning tools are changing the landscape of drug discovery and development. And the recent news about AlphaFold now being publicly available for academic and commercial users gives a quantum leap to the drug discovery processes to predict secondary structures or protein structures, even when the template or the reference structure is not available. So such kind of technological advancement are very welcoming, and it opens new era for revolutionizing the healthcare. And without further ado, I would now hand it over to Ravi for his opening remarks on the behalf of our director.
Good afternoon. It's a great pleasure and honor to invite you all and the many students and my wonderful colleagues. Especially, this is a very timely webinar on AI in drug discovery and research, especially when it is hosted by the hallowed portals of CDRI, which is really very timely. And I can see that I extend a very hearty welcome to all the galaxy of my esteemed colleagues and speakers here. We have a wonderful program. I'm very excited and very keen, and so are all the students and staff and other participants who are now today live watching to hear new inputs and new this thing on different aspects of drug discovery, right from chemistry, computational, and all the other areas. So I request Dr. Gautam Panda to extend welcome to all our speakers and people here.
Good afternoon to all of you. Soon we are going to start the webinar on artificial intelligence on drug discovery research, AI-DDR 2021. This is the first time we are going to organize on this topic. As Niti says, we would have loved to meet all of you and have a very fruitful interaction, but due to this COVID situation, we are not able to do it now. So I, on the behalf of director, as well as on my personal behalf, and also from CDRI fraternity, I welcome all the speakers, Professor G. Narore Sastry, Professor Subramania, Professor R.B. Sunoj, Professor Devapriya Kumar, Professor Sandar Ahmed, to give a talk on this topic. As you can see from the artificial intelligence, it's composed of two words. Artificial means man-made and intelligence means thinking power. So that means artificial intelligence is man-made thinking power. So for the next 3 hours, we'll have a very exhaustive session how to apply this artificial intelligence in drug discovery research. And I hope this will put us a platform to interact with all of you to have some fruitful discussion and collaboration in future. And as Rabi and Niti says, this is just only a beginning. We have many more lectures will be coming in coming future. Thank you all.
Thank you. So we quickly begin with the first speaker. It's none other than the speaker is from CSIR family, Professor G.N. Shastri, who is currently the director for CSIR lab in Jorahat. He completed his education and PhD degree from University of Hyderabad. Then he started as an independent group leader in Pondicherry University and later moved to CSIR-IICT, where he headed the Center for Molecular Modeling. He is recipient of Shanti Swarup Bhatnagar Award, DBT National Bioscience Award, and JC Bose Fellowships, and many other recognitions nationally and internationally. And I hand over to Professor Shastri for his talk on machine learning approaches for drug discovery. Over to you, sir.
Okay, thank you very much. Are you able to see my slides? Hello?
Not yet.
Not yet.
Yeah. I don't think yes. Hello, now?
No.
No, you don't.
Or else Devapriya Kumar are here. Sunoj is there, Sunoj?
Yes, now it's possible to see.
Hello, are you able to hear? Are you able to see the slides? No. Call somebody, Nagamani. Yes.
I think your desktop is being shared, so maybe you have to just focus on your slides. Yeah, it's fine now, I think. If you go full screen, it is fine.
Hello.
Yes, sir.
Yeah, it's
Okay.
fine. It's fine.
I'm sorry about it. So, first of all, I would like to thank Dr. Ravi Shankar and Dr. Gautam Panda, all CDRI team, and Professor Tapas Kundu for giving an opportunity to be present here. I just wanted to kickstart by saying a few things about drug repurposing or machine learning, a little bit, but mostly about the computational modeling. Oh, I shared a wrong. Sorry. Okay, so there was a wrong set of things which are shared. This has to be shared.
I hope it doesn't show you show us the bank PIN code of yours.
Maybe. I really don't know, because I just rushed from the other thing. So essentially, we have drug discovery. We cannot share. So, okay. So I will probably try to do it without slides. So drug discovery is a topic which has started.
So Professor Shastri, your screen is still shared. If you are not using slides, you can stop sharing screen, I think, because we see ourselves only otherwise.
So now it is fine.
Yeah, I think.
So essentially, drug discovery is a field which started as an exploratory mechanism of trying to find out what are all the properties that a molecule will have to be a drug. And in that process, we, about 100 years before, the solubility of a compound was considered as an important criteria to make that this particular molecule can be a drug. And after some time, people have realized that the solubility in water, although it's an important parameter, they also have realized that the solubility in water as well as the organic layer appear to structure, biological structure is usually a protein, or there may be some other receptor, but most of the time it is the protein structure which is there, and the small molecule will go and inhibit this particular structure and elicits biological response. And when you try to look at the efficacy of a molecule where the drug mechanism is not clear, you don't know what is the receptor, but probably a series of compounds elicits the biological response, and then they have shown the biological activity where you can try to get the quantitative structure activity relationships or CSIR activity or analog based approaches which have come into picture. Once a particular molecule has shown some activity, we will try to optimize the properties of these molecules in terms of their absorption, distribution, metabolism, excretion, and most importantly, toxicity, pharmacokinetic and pharmacodynamic properties to optimize the lead compound into something like a clinical candidate. So we have the drug discovery as one phase and drug development as another phase. After all this early stage drug discovery and the preclinical trials, one will submit an investigational new drug application and then go through phase 1, phase 2, phase 3 clinical trials. And then they will have an NDA clearance by Food and Drug Administration in the United States and the corresponding regulatory agencies in other countries. For example, in India, you have DGCA. And these particular compounds, which were approved as drugs, were originally shown to be very efficient against the diseases, but most importantly, they were shown to be non-toxic. They were shown to be at that particular concentration range, do not have any negative impact on the side effects on the healthy patients. So most of the molecules will probably have to meet this criteria, which is what is known as phase 1 clinical trials. And in the phase 2 clinical trials, you will have to see the efficacy of the compounds, because in phase 1 clinical trial, large number of compounds will fail because they are probably showing the side effects. So when a particular compound is known to be an active compound and it got an FDA approval, there is a possibility that it has met a large number of criteria for that particular molecule to be a drug. The question is whether this single molecule can be shown efficacy on a disease which is completely different than for the disease which is approved, that is drug repurposing. And in these processes, people have used different approaches like polypharmacology approach, or a single molecule will be effective not against one target but multiple targets and display efficacy. And there are a number of compounds which were shown to be active against multiple diseases. And it appeared to be a very, very important concept, which is known as drug repurposing. If an approved drug shown to be active against a different diseases, drug repositioning. Now, how do people develop drugs? How do people discover drugs? This should have a basis in terms of computational modeling, where people have to find out that a particular molecule will have to show the efficacy based on the principle. The principle can be, it might have so much of binding against a protein target. And for that, there are methods like docking, which has come. If you know that this is a particular target and you have a small molecule, and you will be able to rank order the efficacy of the small molecule binding affinities. But we all know that the scoring functions that are used in docking are very, very preliminary, and more often than not, you will just get some sort of a number which will show that this particular molecules might have a good fit or not, but the scoring function will have very, very crude approximation to the binding affinities, and that might not have much sense. So you have more sophisticated methods like MMPBSA or MMGBSA, or in some cases, it is also possible to have more rigorous calculations like free energy perturbation method. And in most of the cases, you will be able to get what is the rank order, which molecule will bind to a particular protein target better compared to the others. Whereas if you try to look at a number of small molecules which are showing efficacy against a particular enzymatic assay or whatever in vivo or in vitro model or animal model that one develops, one need to correlate a computational method which will give results which corroborate with the experimental observations. In that way, you can use any method like regression methods, or you can use different methods based on nonlinear approaches or other statistical approaches. There are methods like support vector machines, self-organizing maps, and there are also methods which are based on artificial neural networks or genetic algorithms, which will be able to fit your data based on a training set and develop a model. And the model can be validated by how robust it is statistically on the training set, as well as validating set, which we call it as a test set. And these particular methods have proliferated quite a bit. And we also have a number of methods which have come for screening large databases. These can be based on any criteria like fuzzy logic, and docking appear to be also a very important criteria that people have used. Now, the question is, what is the relationship between the structure of a molecule and its property? For doing that, we have a number of experimental procedures that are available and a number of computational procedures that are available. But however, if you look at the data that is there in chemical space, about a few years before, we have of the order of a few billion molecules as the upper limit for the chemical space. But now the chemical space is anywhere between 10 to the power of 60 to 10 to the power of 180. That is a mind-boggling number. That is several orders of magnitude compared to the entire microorganisms or even the mole of molecule. It's a very, very big number, and one has to somehow have a good way of comprehending how this chemical space can be put into some computational method or a statistical method. And the conventional statistical methods appear to have a huge problem where the volume of the data or the velocity, the 5 V's that are there in big data, the volume itself is very high, but the new molecules that are coming up with new properties, the velocity with which they are adding to the chemical space is also very high. The variety is also very, very high. The variability is also very high. And one probably has been syncing with a lot of data, which is very difficult to fit into the conventional computational modeling. And as one sees that we have the two paradigms, which is experiment and theory, and the modeling has become the third paradigm in computation. And theory has been there for a long time, but computation is also providing the data. So it becomes very difficult for a theory which will also have concepts, rules, hypotheses, or laws to explain the data that is produced either by experiments or by modeling methods. So people have come up with ways of how the data itself can generate new theories. This becomes very, very relevant and important in large number of fields because you have a lot of data-driven theories which are used in our searching in the public domain places like Google or Facebook or many other things. The data-driven approaches or artificial intelligence and machine learning has played a very, very important role. So in that way, when the data is in the numerical form, you will get some information. But there is a lot of data which is also there in terms of text. So the text mining plays a very important role. For example, there may be some data that is available in structured form or not so structured form. The data may be there in textbooks. The data may be there in different languages in the text form. So text mining has become an extremely important area, and there are a number of applications in drug repurposing using text mining. And one has to see that the text mining approaches have played a very, very important role. And I'm sure that we have different ways of depicting a molecule. For example, you have a molecule, and how do you convey the structure of a molecule to the computer? The best way to convey the molecular structure to a computer is to give the three-dimensional coordinates and what are the atoms at what positions and what are the distances, which will be reflected in terms of the bond lengths, bond angles, and dihedral angles. And that is one way of giving. And another way of giving is the XYZ coordinates. The third way is a two-dimensional, three-dimensional structures which can be given in .mol or .sdf formats, which are very effective, where you can put a number of structures where the three-dimensional information can be fed. But the most important formats which can be used are the alphanumerical strings in the form of smiles. Smiles notation and different kinds of smiles notations have played a very important role. But the friendliest way of trying to find out how to represent a molecule is in the form of fingerprints. Because the Boolean logic, if any information has to be given to a computer, ultimately you have to give it in the form of zeros and ones. And this will be very useful to compare how closer are two molecules. For example, if you want to have two molecules and find out which molecules are similar and which molecules are not similar. In that way, similarity indices like Tanimoto or Jacobi, or there are several methods which are there where you will be able to quantitatively say that these molecules are similar to a particular point. This is easiest to subject a qualitative aspect like similarity into a quantitative matter. So there were a lot of new methods which have come, and in this particular area where the chemical space, which can be in different formats, is available for people to mine it and get the information out of it. And there are valuable databases like Chembel databases and other databases where people are trying to incorporate a lot of information about a particular molecule, their biological activity. Because sometimes some very important molecule will have tons of data, and all of that will be useful, and they will be mined from the publications that are there. And a number of people will try to synthesize the molecule and then get the data, and they may not have any idea what are the properties of that. So text mining played a very important role. And this is very different from computational chemistry or the conventional theoretical chemistry based like quantum mechanics or classical mechanics or statistical mechanics based approaches to deal with molecular structure and their function. However, the text mining also play a very, very important role. The next way of getting the information is in terms of the images. Image processing is also a very important role. For example, if you try to take a small molecule and then try to find out what are the polar areas and what is the non-polar area, or you can divide that into hydrogen bonding donating or hydrogen bonding accepting. And now there are a number of other non-covalent interactions. Are there a plethora of non-covalent interactions which are there, starting from halogen bond, cation pi interaction, anion pi interactions, pi interactions, nitrogen bond, chalcogen bond, carbon bond, and there is a large number of interactions which appear to play a very important role. And then we can also look at molecular electrostatic potentials or any other surface properties that can be generated, like van der Waals and other surface properties, where these images can be compared. And more importantly, people have used the image processing for diagnosing various properties that are there for a tumor. For example, whether it is malignant or non-malignant, some questions are being asked, and image processing is very much used in different fields. So I'm very sorry that I'm not able to put our slides, but I just took this opportunity to tell you that drug discovery has been conventionally dominated by natural products chemists for a very long time. And one of the fundamental challenges that people have in drug discovery is to find out what is the biological activity of a good natural product compound. And afterwards, the synthetic analogs play a very important role. And for a long time, chemists and pharmacologists have played a very important role in pushing this drug discovery process. But once people are able to get atomistic understanding of the biomolecules, and when the protein crystallography has come to the fore, and people are able to find out what is the mechanism of action at a molecular level, and also ask questions where people try to find out which particular small molecule bind to the receptor in which particular way, then things were completely different. It was dominated by biologists, where the field like chemical biology has come in a very big way, which is a little bit different from biochemistry. We can think about these aspects. However, there is a bigger question that people are able to ask. Earlier, people are very happy if a particular molecule is biologically active. When it is biologically active, and then when you try to get an FDA approval, this is used. And in most of the cases, the mechanism of action is not even clear. Whereas when chemists and biologists started working very closely with pharmacologists, people are able to not only find out why a particular molecule is, whether the molecule is active or not, but also find out why a particular molecule is active. This becomes very important because in many cases, these drugs will develop resistance. The drugs might develop resistance if it is against viruses due to mutations or bacteria, but they can also develop resistance because of the operation of some efflux proteins. For example, like a P-glycoprotein, or there are different ways in which the drugs can develop resistance. And unless one is able to come up with alternative ways of dealing with this resistance, we will not be able to tackle this challenge of the disease. Whereas this is very, very clear in our current quest of the COVID. We all know that even if somebody develops a drug for a particular strain, if there is a mutation which might be very different and that can be escaping the antiviral activity of a small molecule. So one is forced to understand what is the origin of the, what is the mechanism of action, and then and only then, if the drug develop a resistance, they will be able to come up with newer ways. Having said that, the biological screening of the molecules is running into a few thousands or sometimes even more than tens of thousands to get one molecule to be finally approved by FDA. And most of the time, it is a good idea to explore whether already approved drugs will be able to show efficacy for diseases which are coming newly. The drug repurposing showed extremely good benefits where the machine learning approaches play a very important role. For example, if you have a drug which is already approved, if you want to go for a new indication, you can have a faster route because we all know that at that particular concentration, it will be not toxic. So we have to see that there are some drugs. For example, there are drugs for anti-cancer compounds where they might have some toxicity, but you can reduce the concentration to a very low level. Maybe at a different concentration, it might become an antiviral compound. And there are other compounds which are antibacterial, which might be antiviral. And our ongoing work, actually some of them are under the press and some of them are not yet published. And we are now looking at the entire drug bank and try to find out whether we will have some indications that these molecules can become antiviral, or these molecules can probably interrupt the protein. Hello, am I audible?
Yes, sir. Yes, we can hear you.
So I know that many experts are following me, and a number of techniques are available now in artificial intelligence and machine learning, which can be applied. We have used some simple techniques like XGBoost and a few other things where we are getting much better correlations compared to support vector machines or random forests or the other neural network methods which are there. Because when the data is huge, when you are running into a statistical equations which has more than 10,000 data points, many of the conventional methods will have a problem. For example, if you have a linear regression approach or even multilinear regression approach, you may want to really have a way of, you may want to probably have a reason to get a particular classification or optimization or prediction. But if you go for neural networks or deep neural networks, you can try to optimize the various parameters just to get your final results. Many times people say that this is like a black box, and it is a black box because the whole idea is to make a model which is very robust. Sometimes some of the people used to say that, oh, this particular molecule is active. Why it is active? Can we understand? Sometimes we may not have to understand. That is the basic difference between theory or even second paradigm, that is a computational modeling, to data science. Data science is with the principle that only data speaks. We don't know what is happening. Why, for example, if the pandemic is spreading, why it is spreading so much in Indonesia and Singapore you can take, and Brazil you can take, and you will get completely contrasting pictures. Turkey, it gives a completely contrasting picture. What is the rate of infectivity? What is the rate of transmission? What is the rate of mortality? It's very different. So whether this will involve reporting or underreporting is not very clear. So for every complex data, you come up with a theory. And how do you get the theory? You will just give all the parameters that are there and then come up with a model, and then this will give a theory. So
Dr. Sastry, please try to summarize.
Yeah, sure. So what we will have to look at is, when you try to do the drug discovery, instead of the new chemical entities, drug repurposing is one approach which one has to use. And in that drug repurposing, while the computational approaches based on the structure-based, analog-based, pharmacophore-based approaches are very much there, the approaches based on machine learning are here to stay, and we will have a lot of new things that are coming up where the chemoinformatics and bioinformatics, the best way to integrate them is using machine learning approaches. With this, I thank the organizers for giving me the opportunity, and I'm very sorry that I'm not able to. I had some problem with some file which got corrupted at the last moment. Okay, Gautam.
Yeah, thank you.
Thank you, Dr. You have rightly mentioned. Thank you, Dr. Shastri. You have rightly mentioned that the conventional drug discovery and development approaches are very, very time and cost intensive, and AI-based approaches would definitely accelerate this this process. And especially there's vast data available, both clinical data as well as the toxicity and efficacy data. So it becomes easier to use these approaches and utilize them for different drug combinations and also in personalized medicine. If I may ask a question that AI-based programs, there are no formal education of AI-based programs at university levels or at master's level, which can, give hands-on trainings to people, and then they can build on that. So, any comments in this line?
Yeah, thank you very much. It's a very important point. First of all, I feel that experimental data, theory, and modeling, these three are essential, and the data that is generated from that requires something beyond these three paradigms, and the fourth paradigm have come. So there has to be a very effective interplay between these four paradigms of science to address questions of profound importance. Regarding the question of how do we address this lacuna in our pedagogy that we don't have this AI? And definitely this has to be taken up, and efforts have to be made. As our education system, we paid a lot of big price where mathematics and especially statistics, when it was taken out from medicine and biology. So we have to give due importance to statistics and biostatistics, where everybody who is in life sciences and medicine know. And once we give due importance to statistics, the artificial intelligence and machine learning are a slightly higher or big cousins of these two methods. And then these methods have to be incorporated, like supervised learning, unsupervised learning, and other methods have to be incorporated. And I'm sure that even at a +2 level, these will definitely be incorporated, because everybody is talking about it. And not only for drug discovery, for any branch of science or engineering or even geoscience or physics, we cannot really ignore machine learning and artificial intelligence. This is a bigger need. I think people will realize sooner or later, countries will start adopting.
Okay.
Thank you very much, and thank you for sharing your thoughts. And we move on to the next speaker, Professor Subramania. He is also a known name to CDRI family, and he's a structural biologist by training, and he received his PhD from ISC. He did his postdoc from University of Oxford, and he was in CDRI from 1998 to 2002, and then he moved to Origin Discovery Technology. He's currently the director of IBAB and a Biocon chair. And today he's going to talk about AI and machine learning in drug discovery opportunities and challenges. Over to you, Dr. Subramanian.
Can I just interrupt and request? Can you mute all the participants and unmute them only when they have to speak? Because sometimes there is a disturbance coming from you.
It's okay. Thank you very much. Am I audible?
Hello? Yes, you are audible. Thank you.
Okay, thank you very much for this kind introduction, and also this opportunity to speak in this webinar session. Of course, I'm very proud of being part of CDRI at the beginning of my career. I said 1998 to 2002. I still cherish my memories of staying in Chetar Manzil and traveling to Aliganj and various parts of Lucknow city. Of course,
sir, you're not visible. Only you're too close to camera, I think.
Okay, I have a specific.
Yeah, this guy. Thank you.
I have a special problem. My voice is very low. Therefore, I speak very close to the computer.
It looks very good.
And then obviously, I will lose my picture. Sorry about it. So thank you very much for inviting me for this webinar, and thanks to the organizers, Ravi, Gautam, and Tapas for this invitation. So let me start with some introduction into the application of AI in drug discovery. I don't know, Gautam, I sent you my slides. Should I project it myself or?
I think you can do by yourself.
Let me see. Is it visible?
Yes, sir. Go ahead.
Okay. Okay, hope that my slide is visible now. Honestly, personally, I haven't really done much in the space of artificial intelligence in drug discovery area. Although,
Subramanya,
yes,
actually, you can put your slides in the slide mode.
Yeah, I will do that. I haven't yet started that.
Okay. Okay.
Okay.
Yeah,
So, however, I worked in pharma industry for a reasonably long period of time, and therefore, I have put in some efforts in the traditional methods of drug discovery, and therefore I can probably compare, the difficulties or challenges faced by the traditional drug discovery methods and how, artificial intelligence can help at least to alleviate some of those bottlenecks. Of course, I have put in efforts in terms of bioinformatics or literature work on artificial intelligence in drug discovery. So let us start with some of the challenges that the drug in the process of drug discovery and development. Obviously everybody knows this. There is a formidable challenge in terms of the timelines for development of the drugs, 10 to 15 years, and costs, varied estimates, but it could be up to $1 billion. But honestly, a lot of this effort and money is actually going down the drain. This is because only 1 in 10 projects that you have started with will probably reach even the stage of clinical trials. And even after entering clinical trials, only 1 in 10 might succeed. So that means the cost of drug discovery is actually cost of failure. And there are many disease conditions, cannot be really addressed through traditional drug development processes. The target-driven method that we have been using in the traditional drug discovery will work well if the target is really druggable and you have a good biological and functional understanding of the target. Many of the receptors and membrane proteins still remain out of bounds of the drug discovery effort. It's also very limited due to the complex nature of cellular interactions. That means, there are so many interactions that are happening within the cell with this particular target protein. How they all influence this particular target is unknown. And also there are a lot of cellular pathways, feedbacks, which we don't fully understand. And that's why, traditional methods, many times it's very hard to design the drugs. So why is it so challenging is that, many of these drugs, I will only focus on the small molecule discovery at the moment, are chemically synthesized compounds, that can specifically bind to a target involved in the disease. There are large chemical libraries may be available, either physical or virtual, and it can be screened to identify some potential hit or drug candidates. Then these compounds will have to go through numerous rounds of in vitro as well as in vivo tests to develop this into a promising candidate. Of course, the rational structure-based drug design or other rational approaches such as CSIR can be used, which can help avoid some of the initial screening stages. But still, it won't replace a chemist for sure. It still requires designing, synthesizing, and evaluating a large number of compounds. This process of optimizing the compound into drug candidate is very expensive and time-consuming. It's not really easy to predict which chemical structures will have both the desired on-target activity, which we call biological effects, and the properties that are needed to become an effective drug, such as physicochemical properties, pharmacokinetic properties, and various other properties that makes it a good drug. Even if you have all these things, clinical translation is still very challenging. It's very hard to predict. So it's clear that something has to be done very differently. So question is, what can be done differently? In order to look into that, we'll see what has changed. There are a lot of technological advancements happened over the past couple of decades, which have led to massive amounts of data generation and digitization as well. Vast amounts of computational resources are also available in terms of the processor capacities, data storage, cloud computing, technology related to that, etc. There have been very sophisticated algorithms that have been developed in the past couple of decades in the areas of pattern recognition, machine learning, artificial neural networks, and so on. Of course, with all the incremental knowledge, we understand the diseases and biology definitely much better. That is, that a knowledge increment has really happened. Can we now use this is the question. Let us look into the biological data. Biology is definitely become data rich. There is massively parallel data generation that's happening. It's coming from genome sequencing, high throughput drug screening, microarray, combinatorial synthesis, shotgun mutagenesis, protein structures, proteomics, and 500,000 biomedical articles per year, which is getting digitized now. However, there is a problem with this data. This biological data is certainly very noisy and with little or no theory at all. And data is extremely heterogeneous. And a lot of scales are applied to this data. Therefore, it is with multiple scales. And many times this data is highly specialized. Therefore, there is no easy way of interpreting this data. So it needs robust analysis methods, statistics, cross-scale and heterogeneous data integration, which requires an intelligent system to analyze this data and interpret this data. If you just look at the growth of biological data, this is just a chart to show, see the number of entries that are coming into the gene bank or the PDB databases or the biomedical data in terms of PubMed searches. Therefore, there is a lot of data that's being generated, and intelligent systems are well suited to analyze the complex and heterogeneous data from biology and medicine. This is where the AI actually comes into picture, really. Okay. So the unparalleled data processing potential of AI systems, they can accelerate and reduce the cost of discovering new drugs. There is massive quantities of data. It comes with its own challenge of acquiring, scrutinizing, and applying that knowledge to solve complex clinical problems. Certainly, it motivates to use AI because it can handle large volumes of data with enhanced automation and its own decision making. So AI is a technology which is based, when it's a technology based system, it involves various advanced tools and networks that can potentially mimic human intelligence. It also utilizes systems and softwares that can interpret and learn from the input data to make independent decisions for accomplishing very specific tasks. So the question really comes here will be that, is it something new? I don't think it is really that new, so to say. Now, if you look at computational approaches to drug design, it goes back to at least to 1970s, when the efforts were focused on understanding the structure-activity relationship of molecules to help suggest new candidates. Definitely the application of CAD in drug discovery dates back to several decades before. In fact, at one point in time, there was also such a big hype about CAD that, there may not be, scientists may not be really needed anymore, to at least to the design of compounds. It was really hyped to that stage. And of course, computational approaches are routinely used to screen virtual libraries of compounds using structure-based or ligand-based approaches. So the use of computers in a general sense for design and development of drugs was already there for several decades too. Is AI new? I don't think even AI is really new. So you can see that, the initial efforts, or at least the idea of artificial intelligence, goes back to 1950s. In fact, it goes back to World War II, when the German Enigma machine codes were decoded by Alan Turing and group. That's when people started thinking about building intelligent machines that could sense, reason, think like people. It's a concept of general AI. Certainly, it's likely to remain in the science fiction books for some time to come. It's not really a reality at this point. However, the rapid growth in computer processing power and the availability of large data sets and the development of advanced algorithms have driven major improvements in one particular area, which is machine learning. This has actually helped to bring about the so-called concept of narrow AIs. We also call it weak AI, which focuses on specific tasks. It's not like it can do, everything from smelling to eating, but it can do very specific tasks, which is a narrow application of AI, sometimes also called domain-specific AI applications. These include, for example, improved abilities like natural language processing, artificial neural networks designed to mimic the way brains make sense of a particular feature of the world, and so on. Such techniques are already in widespread use in many fields, such as computer vision or voice analysis, even, probably our Google Maps in terms of route selection and so on. It is this domain-specific AI which is really helpful, and people are actually borrowing from other domains to use in the area of drug discovery. And this has led to a wave of startup companies. Currently there are at least 230 startup companies which is employing artificial intelligence for drug discovery. Now, if you look at what is there inside artificial intelligence, this is just to give an idea. At the core of AI in the drug discovery process is actually the learning process. There has been tremendous advancements which have been made in the learning process. However, the majority of learning tasks or techniques used in drug discovery belong to these 7 categories: the supervised, unsupervised learnings, semi-supervised learnings, active and reinforcement learnings, transfer and multitask learning. These are all mathematical principles, basically. How do you relate a given set of data and labels, that means properties or features, to another set of data which does not have such kind of labels? So we'll have to find out a mathematical relationship between one which has a label and one which does not have a label. And in more advanced machine learning, such as reinforcement or active learning, you will also try to get the feedback. Of course, when you learn for the first time and try to apply it, there could be mistakes or misrepresentation. Now you go back and improve the training set, which is a sort of active learning process. That means just like the human brain does errors, and from the mistakes we learn, the reinforcement and active learning processes will do exactly the same thing. They will go back and improve the training model whenever there is an issue with the prediction that's made. So this is the core of the AI in drug discovery, is what sort of machine learning is suitable for what sort of processes. How do we really apply for a given data set? So that's where everything lies in the concept of AI in drug discovery. Of course, there are algorithms which are associated. That means the computer programs which carry the mathematical principle in learning, as we saw in the last slide. There are many learning processes. There has to be algorithms which will translate that mathematics into computer programs, and these are the algorithms. There are many types of algorithms which are available. Artificial neural networks. There are many different types of algorithms available within artificial neural networks. There is decision tree kind of algorithms such as random forest. There are instance-based algorithms also, such as the KNN or K nearest neighbor models and so on. different types of algorithms will be very useful for different types of processes, depending upon the data that you have in hand and the problem that you are trying to address. So if you look at this and the algorithms of artificial intelligence into drug discovery process, you will see that it can be applied to multiple areas of drug discovery to alleviate some of the bottlenecks in the current process. It can really apply to most of the areas of drug discovery, provided there is data available. So I will highlight a few of the areas, not by any means to cover extensively every process in drug discovery, but at least some areas which are commonly used in drug discovery process, and see what has happened in those areas with the application of artificial intelligence. So virtual screening is one of the very early steps in any drug discovery process. It has been employed routinely, majorly for reducing the cost of the high throughput screening, and also to some extent increasing its efficacy and predictability. The machine learning models implementing aspects of AI methods have been successfully applied in several aspects of virtual screening pipeline. One of the popular ways you do the virtual screening in the so-called structure-based virtual screening is using the docking techniques. Anybody who has used docking, I'm pretty sure, is aware of the fact that the major limiting factor is the reliability of scoring functions. You dock it, you can virtually dock any compound you want to any protein structure you like. It will always produce some score. The question is, what would you believe? So there is ambiguity and question of reliability of scoring functions while using any of the docking programs, and therefore it's very hard to prioritize compounds based only on the docking scores. There have been a lot of efforts made to develop data-driven scoring functions based on data mining, knowledge-based approaches, machine learning approaches, for deriving better scoring functions for the structure-based virtual screening. One of the issues why the scoring function or docking score functions are like that is because they are all predetermined. That means either they use the force fields which are predetermined, or they will use empirical energy models, which again has rigidity in terms of predetermined values. The question then is, can we really use non-predetermined scoring functions, which will learn from the data themselves in a dynamic fashion, rather than saying that this is the force field or that is the energy? You learn what is the energy and what is the force field. So such kind of scoring functions have shown to be far more reliable as compared to traditional scoring functions. We can see AI-based non-predetermined scoring functions such as SVM-based score or random forest-based RF score or artificial neural network-based NN scores. They have shown far better accuracy as compared to the traditional methods of scoring functions. Similarly, another drawback is about visual examination of docked poses. Since we can't really trust or rely solely on the docking score, so we have to really manually see where is it binding, how is it binding, what sort of interaction it makes, and so on, which is very time consuming. You can't keep on, for every molecule, watch how it's working. Is there methods that can, remove this manual intervention? That's where the deep learning methods extract the features and learn from features in a data-driven way without much human participation. There is one tool called DeepVS, for example, which is one such model to improve docking based on docking-based virtual screening without much manual intervention. In a very recent benchmark study, you can see DeepVS actually outperformed all other scoring functions or virtual screening tools when it is used for docking against 40 receptors using 2,950 ligands. This was the best performer in that benchmark study. Another early success of application of AI is in the area of protein structure prediction. You know about the AlphaFold, the AI subsidiary DeepMind, of the Google actually, announced that its AlphaFold program now can predict protein structures which are approaching the quality of experimental protein structures such as determined from X-ray crystallography. That's the level of phenomenal success that has been achieved in terms of structure prediction. Obviously, ability to predict the structures would substantially accelerate the structure-based virtual screening, because now we are limited by only those targets for which there are structures available if you want to apply structure-based virtual screening. This is just a schematic representation of the architecture of this DeepVS program. It takes the output from the docking program to begin with, and it extracts features from structural data such as distances, atom types, partial charges, and distinguish between actives, decoys. Then it actually runs through a convolutional network, which extracts further, deeply hidden information, and thereby really gives an output which is far more reliable. If you look at other methods, the QSIR is one of the very popular methods which has been used traditionally by a lot of medicinal chemists to understand the relationship between structure and the biological activity. Of course, it is also used for ligand-based virtual screening, which is based on the hypothesis that structurally similar compounds will have similar biological activities. There have been a lot of efforts that have focused on implementing AI algorithms in QSIR models now, such as the random forest-based approaches, or producing approaches and other methods. They produce relatively reliable prediction performance. Another big change or big improvement that has happened is actually in the multitask QSIR. What happens is in traditional QSIR, it's a one-to-one relationship. You have a structure and you have a biological activity or target-based activity, and you try to identify new compounds which are similar to these compounds and predict what could be the activity of such a compound. However, the information available with chemical compounds could go far beyond just the target-based biochemical activity. It can have, for example, data on physicochemical properties of the compound. It may have, data on the pharmacokinetics. It may have data on the permeability. It may have data on the toxicity, and so on. A lot of additional data is available, which is associated with the compound libraries. How do we really use it? Instead of saying that one particular compound is likely to have an activity against this particular target, can it really select a compound with optimal properties, such as optimal physicochemical properties as well as biological data, which is actually multitasking? Okay, so such kind of QSIR models can classify compounds based not only on their target activity, but also on other experimental information that's available. So this is just an example of the multitask QSIR models which have been developed using the AI methods. As you can see, there are a lot of data available here, different types of experimental data, starting from the biochemical data to cell-based data, protein structures, various assays, and as well as different kind of models. So you can retrieve this initial data set from a database like Campbell with one or more experimental data. Then you can start retrieving or creating topological descriptors. Instead of normal descriptors, we create topological descriptors, which are sort of bond-based, which connects multiple experimental data with a single point as a quadratic indices are calculated. And the best performing multitask quantitative structure biological effect relationship model can be applied to screen either fragment libraries or virtual screening. As an example, in this particular paper, they have screened to find out compounds which have potential hepatitis C activity with very high degree of predictability, which they have demonstrated using experimental studies. Another area which is very important in the drug discovery space is actually the physicochemical properties and their relationship with the biological behavior. This is just a schematic representation. There are many properties such as solubility or partition coefficient, pKa's, stability, molecular weight, so on. So they would have influence on systemic circulation, excretion, metabolism, toxicity, pharmacologic effect, brain permeation, and so on. If you look at the application.
You have 4 minutes.
Hello?
Dr. Subramania, you have 4 more minutes.
Right. So if you look at the application of AI in predicting these ADME properties, again, there are quite a number of programs that have been developed, which are now available to guide the synthesis of the molecules and can also minimize issues later that can arise because of non-optimized ADME properties. There are many AI-based programs. One such program is ALOG-PS, which is based on the associative neural network, which is also called ANN, that combines the elements of the feed-forward network with the k-nearest neighbor approaches, which has predicted the log P values reasonably accurately, at least for low molecular weight compounds. Similarly, metabolism is another issue, which mainly happens because of the cytochrome P450 isoforms and the UDP glucuronal cell transferase, or UGTs, in the manner of phase 1, phase 2 metabolism. Okay, so again, if you have an ability to predict the site of metabolism while synthesizing the compounds, that will greatly help in the synthesis of or design and synthesis of compounds. Now there are several AI-based modules which are available. For example, RS Predictor is one such program which attempts to identify potential sites of CYP-mediated metabolism of molecules. It uses a combination of hierarchical, quantum chemical, and topological descriptors. Xenosite is another one which is based on the neural networks. Predicting toxicity is another major challenge. You can see that a lot of compounds will just enter the clinical trials and fail because of toxicity. There are, assays available, high throughput assays. They're expensive, and also their reliability is a question. So if there is computational models that are available for rapid, inexpensive, and reliable alternative, then they will be of high demand. There are many web-based tools that are available, such as LimTox, or PKCSM, ADME-SAR, ToxTree, and so on. And advanced AI algorithms have opened up new avenues for predicting toxicity, such as predicting the toxicological endpoints or classifying toxic compounds, and various types of methods have been used, such as graph-based methods. This is just a slide to show that there was one particular competition which is called the Tox21 Challenge. In this, it is actually organized by NIH, EPA, and US FDA to evaluate the computational techniques to forecast the toxicity of 12,707 experimental or environmental compounds and drugs for 12 different toxic effects. The ML program, which is known as DeepTox, really outperformed every other program in the available in the market, and this is really based on the deep learning technique using the neural networks. This is just the outcome of that to predict the toxicophores. The DeepTox has predicted these toxicophores from multiple layers. As you can see, some of them chemists will immediately recognize. Some of them are hidden, which is not so easy to recognize either. Finally, the last area where it can make a substantial impact is in drug repurposing. There are conventional methods which have been used based on the chemical similarity or molecular docking. Now it has completely, or it is shifting more and more towards systems biology approaches, which utilizes gene expression data, phenotype, side effect based approaches, discovery of new potential indications based on the genome-wide association studies, or based on the disease network-based approaches, such as using the CAC pathways or Reactome or such kind of public databases that are available. Now, with the availability of this vast amount of chemical and genomic data, as you can see, this is also very heterogeneous data. Of course, AI really is the competent platform to approach this problem. There are various AI-based methods such as Predict, SLAMS, and other programs like NetLab RS and DTI-Net have been developed to identify possible drug repurposing candidates. This is one such approach, for the drug repurposing, different types of processes and data, and the green boxes are data and the red boxes are algorithms or processes. Using these methods, there are many drugs which have been now proposed, at least in the context of the COVID pandemic or against the SARS-CoV-2 virus. Just to give some few early success stories, there are 2 or 3 I will give, and then I will conclude. In April 2021, there is this German biotech company called Evotec, which announced a phase 1 clinical trial for a new anti-cancer molecule, which was created in partnership with a startup company called Exscientia, a company which is based in Oxford, UK, that applies the AI techniques to small molecule discovery. The target was actually a A2 receptor antagonist, which is developed or designed to help T cells fight solid tumors. It's an immuno-oncology target. It was discovered in about 8 months by harnessing the Centaur Chemist platform of this Exscientia, whereas traditional processes usually would take about 4 to 5 years. This AI platform actually can computationally screen millions of potential small molecules, compare with various properties to identify lead compounds. The lead compound they have identified shows some of the best-in-class characteristics. If you look at another company called Benevolent AI, there was a collaboration launched between AstraZeneca and Benevolent AI to discover new drugs for chronic kidney disease as well as the idiopathic pulmonary fibrosis. It's a serious lung fibrosis issue. Using knowledge graphs, the Benevolent AI could come up with a target utilizing some data that was mined from the literature, using natural language processing, some produced by chemical or pharmacological experiments, some arising from gene studies and so on. They were able to identify a drug target, which was subsequently validated by AstraZeneca. Now that is going to enter into clinical trials. Another company is, Cyclica, which is a Toronto-based company. Again, they have a different kind of an approach utilizing the polypharmacology, which is based on protein-protein interactions. Using that kind of an approach, they are really able to screen a large number of compounds. In fact, their performance outperformed the mass spectrometry based approaches for correctly identifying off-target drug interactions for small molecules at a fraction of time and cost. Now they have partnered with over 100 global pharma and biotech companies as well as academia in various areas of different diseases. This is just to highlight that there is an explosion of sort of collaborations that are happening in this area, both within industry, between industry and academia, as well as academia and academia. And therefore, there is a lot of potential in this area as of now. Finally, AI, in particular, I would say deep learning techniques, which utilizes the neural networks, has the potential to impact multiple fields of drug discovery. Although it is not really a panacea, don't think that it is going to be a magic bullet, that will find the drugs tomorrow. That's not going to happen. It's not really a solution for all the problems of drug discovery. Application of AI can increase efficiency in some parts of the drug discovery pipeline, and it will certainly advance innovative drug research and development. However, its application is really dependent on the quality and quantity of data, and it can only be as good as the algorithm that is being developed. It can certainly assist scientists in their various roles in different disciplines or specialities of drug discovery and development process. But if you are a seasoned scientist, I'm sure that there is something to learn from this application. The applications of domain-specific AI in industry is just beginning, and it will definitely take some time to have a substantial impact in the process itself. Till then, drug discovery will have to remain a slow business with a careful management of risk within the constraints of responsibility to treat patients and these shareholders' interests and so on. So it will continue that way for some time, although there will be local and domain-specific applications of AI which can help. So that's my presentation. Thank you all for patiently listening.
Thanks a lot, Dr. Subramania, for such a comprehensive talk. And if I may ask this question, that you already have an experience of working in both academia as well as in pharma sector. So if we look at the Indian context, we don't have too many companies using AI-based approaches, because for Indian population, we don't have a very exhaustive quality data wherein we can do these predictions of toxicity, pharmacokinetics, or such kind of predictions. So as you have rightly pointed out towards the end of your presentation, that it will take time to kickstart. But how can academia and industry join hands for generating quality data and doing more accurate AI-based predictions? Because ultimately they need thorough validation. If we are not able to validate the AI-based predictions in Indian context, in our population, then we will not be able to accelerate the drug development initiatives which we are trying in India. So what are your thoughts over it?
You're absolutely right when you speak about the Indian context in terms of utilizing the artificial intelligence. In fact, forget about artificial intelligence. There is very few companies within India which are really involved in novel or new drug discovery efforts. There are very few you can count.
Yes.
This is because of various reasons, both cultural, economical, and social reasons. We have been brought up with more like generic manufacture rather than discovering new drugs. Having said that, it has now you can see actually a wave of startup companies, because I'm also associated with a lot of incubation centers at our institute. We also incubate many companies. I do see large number of startup companies which have started in this direction of using the AI-based approaches in drug discovery, although many of them are more involved in development of platforms rather than using it for drug discovery. But obviously the world is changing. It can't remain the same. You may be slow, but one day you will have to adapt. So therefore, I think time is coming up in that direction. There are more and more companies which are now putting together some efforts in that direction. And of course, there are multinational pharma companies which are always interested in collaborating with academia, even in India. So that will be one way out. As far as the data, whatever you said is concerned, of course, there is some data which is India specific. We may not really have very high quality data, but data is also available in public platforms. For example, the data related to chemical compounds. No, that really is not population specific or country specific. As well as, as long as high quality data is available, you will be able to use. There are definitely a lot of public sort of efforts from various organizations which are really putting a lot of money, such as, for example, the Microsoft or even the Google and other places which are putting in a lot of money to develop such kind of databases which may be accessible for all the scientists. And hence we may be able to use that kind of public domain data. Although generating high quality India specific data is very important, it will take some time to develop. Hopefully, at least there could be academia interactions.
Yes,
which can happen.
definitely.
Okay.
Okay. Thank you so much for answering to these queries and highlighting both the opportunities as well as the challenges associated with AI-based approaches. We move on to the next speaker, that is Professor R.B. Sunoj. He is the professor at IIT Bombay. Is Professor Sunoj already there?
Yes.
Yes, we welcome you on the behalf of CDRI family. I would like to introduce Professor Sunoj to the audience. Professor Sunoj did his PhD from ISC, and he's currently heading his research group at IIT Bombay. He's recipient of INSA Young Scientist Medal and also Shanti Swarup Bhatnagar Awards. He's done a lot of seminal work in the area of quantum mechanical analysis of transition states. And he is the chemist who's going to talk about how molecular vibrations and electronic transition would help in designing new and improved catalytic transformations and how they are important for pharmaceutically important compounds. So over to you, Professor Sunoj.
Thank you for the kind words of introduction. First of all, Dr. Panda and others for giving me this opportunity to share some of the work that we do in the AI as applied to chemical space. So I'm trying to share the slide, and as usual, there are some challenges. It's not getting loaded.
Should we share it from our end, and you can just mention next, and we can. Should we share it from our end? Are the slides?
Dr. Sunoj, we have your slides.
No, I have not sent.
Okay, you have not sent it yet. Okay, actually, you told that you can share from your end itself.
Yeah. So this. Whenever I use Microsoft Teams, there is an issue of.
Okay, so what you do, you can send your slides to my email.
Okay.
So that I can operate from here itself.
Sure. I'm doing it in a minute.
Okay.
So I will continue to give introductory remarks.
Yeah, sure.
Okay. So what we are trying to accomplish here in our research group is to use available limited data. So I must underline this particular aspect of what is that we are focusing on. Chemistry has a lot of data in some sense. At the same time, chemistry in the developmental stages, for example, if you are developing a catalytic process, chemistry may not have as much data as required to be able to make best use of the artificial intelligence of the tools that one would have access to. So the question is, can we make use of AI tools for problems which do not have terribly large amount of data, and still we try to customize it? So it would appear as though we are trying to violate the premise of data-based learning process. But that is not the end of the story. We wish to highlight some of the success stories and some of the ongoing activities in the AI ML front. So I've just sent the email to you, Dr. Panda.
Okay, just a moment, open. And I have not received yet.
Refresh.
No. So you can go to inbox and do the refresh.
Maybe there would be a delay. Yeah, you can continue, please. Maybe it will take a few seconds.
I replied to your earlier mail.
Yes, okay. So you have sent to my CDRI account, right?
Yes.
Okay.
So it's about a 6 MB file.
Okay. Sometimes it takes some time to receive mail. Okay, so you can go ahead with the introduction.
Okay, so let me in parallel also try, maybe I will send a link from where you can probably download just as a backup. Okay, hopefully let the slides be ready. So what we, as I said.
So you can type it in the chat box. We can download from the link.
Yeah, so first I have to generate that link and then I will,
Okay,
yeah,
okay,
just
okay.
1 second, because these are unexpected situations.
Yeah, no problem.
Share the link and it has to be for, I'm sorry about this mess up.
Choose. Do you have a Gmail account also, Dr. Panda? So maybe he can send it to the Gmail account. That's pretty faster.
Just give me a second.
Your presentation to my Gmail account also.
Oh, should I?
Yeah. So that
Okay.
is
Scientist Medal, IYBA, and JSPS Fellowship. His area of interest is computational chemistry, and which he comprehensively utilizes to investigate biomolecular interactions of nucleic acids and proteins. And with this, I request Professor Deva Priyakumar to deliver his talk on artificial intelligence and machine learning for drug design. Over to you, Professor Priyakumar.
Yeah, so thank you very much. Thank you very much for your kind words, and also thank you for your kind invitation and for having me. So I will. So as the title says, I will mostly talk about how AI and ML can possibly be used in drug design. Dr. Narahari Shastri and Dr. Subramania gave a nice introduction on how the AI methods in general can make a big difference in terms of making the drug discovery pipeline more accurate, possibly more efficient. So, in general, in the last few years, we have been, using AI methods in general to look at, to study chemical and biological systems, and also into healthcare related areas. Okay, so I thought, there are a lot of students. I thought I will just spend, 1 minute on what is the fundamental difference between the traditional computations that we do and the machine learning way of doing things, right? So, for example, if we take computational chemistry or computational biology, so what we basically do is we have a program or a software that has been written based on principles of physics. It could be quantum mechanics, classical mechanics, statistical mechanics, or it could be, shape-based docking or energy-based docking. It could be anything, right? And then we have this program installed on the computer, and with respect to the inputs that we provide, we get outputs or desired outputs. It could be energies of molecules, or it could be binding affinities, or it could be toxicity predictions. It could be any output that we want, right? So the fundamental difference between the traditional computations and machine learning is that we have simply swapped the output and the program, right? So basically what we do is we have some known outputs for a given set of inputs, and what we are trying to do is ask the computer to learn from the input and output pairs and then give us a program, right? The computer writes the program for us, so that, if I give a certain input, I get the corresponding output. So that's the, fundamental difference. Of course, this is a very simple picture, and this is a simple supervised learning approach, right? So I do not want to spend too much time on this one, but I will show this particular slide, right? So when we talk about from now on, what I will do is I will try to give some context of why AI ML for the drug discovery process. And then I will try to give some examples again at a reasonably higher level. I will not go into the details, but give examples of different tasks in this drug discovery pipeline that ML can be applied from our own work. Okay, so when we take this drug discovery process, when we go from left to the right, we do know that it involves a large number of experts from very diverse disciplines, and this has to, this takes Professor Subramania explained, how this is a very long drawn process and how expensive it is and so on. Okay, so, this particular, I will just show some papers that have appeared in just the last 2 to 3 years. Okay, so for example, the first one, right? So, this paper talks about taking the biological networks. For example, let us say we have a new disease, for example, the COVID-19, right? So how does, for example, the human system and the viral system interact, and how the viral replication process happens? It does involve a large number of biological pathways. Okay, so, let us say I have understood these pathways, and now I want to identify a target. So basically, it takes this information, and then there is an approach which can tell us what are the possible targets for this particular disease that we can use. And then if you take the second paper, so this again, Dr. Subramanian mentioned, it's called the AlphaFold. There's recently another AlphaFold 2 came out, and then again, a lot of news is made about this because, it does much better than most other methods that exist in terms of predicting protein structures just from the amino acid sequence, right? So basically, once we know the target, we know the amino acid sequence, and then we give the sequence, and this particular software or program algorithm can give us the three-dimensional structure. And then this particular paper talks about taking a protein structure and then generating molecules that could possibly bind to this particular protein. And then this particular paper talks about taking these new molecules that are being generated and suggests us ways of how to synthesize, right? And then there is this one talks about robotic platforms that can actually do the synthesis for us, and then doing the characterization and so on. There are several other examples. I'm not going to crowd all of them, but the point that I'm making is, if you look at all these works that have been done in the very recent past, is that we have data-driven methods that is contributing to the entire pipeline of drug discovery, not just in identifying a molecule, right? It's complete. It's just one discipline. The only thing that is common among all these works is that all of them are data-driven. Okay, so coming to that, of course, we are going to concentrate more on the design process, or the somewhere in the middle of this particular whole pipeline, right? So this application of artificial intelligence methods to molecular design actually is not very new, right? So the first paper applying AI or the ML-based methods to drug discovery or drug design problem was in 1970. Actually, I could not get that paper, so I have the second paper that appeared, which talks about using a simple multi-layer perceptron to classify a set of dioxanes into whether they are pharmacologically active or not, right? But then this remained, in the last several decades, this remained a very niche area. That is very evident from here. If you look at, for example, the number of papers that came out just in the ACS journals with machine learning as the phrase in the articles, you can see the kind of surge that's happening in the activities in this area, where machine learning is applied not only in applied research, right, but also in fundamental research. And again, the number of startups that are booming in this space, where AI is applied for drug discovery or in general natural sciences. Okay, so now I will go back to this particular picture, and it's the same pipeline that I'm showing. And what is given in the green are some of the tasks. I mean, not all of them, but some of the tasks that are absolutely necessary to be performed during this particular cycle. So in the context of this particular pipeline, I'm going to discuss some of them, right? So the ones in the red which just appeared are some of the work that we have done, some of them published and some of them in pipeline and so on. I'm just going to take some examples and then show how these modern machine learning methods have started contributing, in this particular, drug discovery process. Okay, so, so if you, when we go from left to the right, of course, I'm not talking about the biology part. Let us assume we have identified a target, and then we have a structure of the target, right? So the next step is to identify a binding pocket, right? So how do we normally do? We could use different kinds of methods, like based on experimental information or based on geometry, like the 3D geometry, and then energy-based descriptors and so on, right? So one of the philosophies that we try to apply is to see if we can use the existing knowledge, right? So it's not, we don't have to necessarily start from scratch, right? Today, we have large number of solutions to these tasks already exist, right? What we want to make, what we want to do is to actually make it, more efficient or, more accurate. So that's exactly what we do. Let us say I have a protein structure, and then what I want to do is to identify the site where the ligand can bind and possibly intervene the functional part of that protein or functional aspects of the protein, right? So, for example, F-pocket is a method that has been existing for a really long, period, right? So basically, what we do is we consider this, the F-pocket. What F-pocket does is it considers protein as a three-dimensional object, and then identifies all possible pockets just based on geometry, right? No biological or chemical information is there. So now what we want to do is, this is comprehensive. Why is it comprehensive? Because it is shape based, and then we know that the binding is dependent on the shape of the pocket in which the ligand has to bind to, right? So now what we do is we just take all these pockets and then use a conventional neural network and then see if we can use the modern machine learning method along with the existing method, and then see if we can do much better. And that's exactly what we see. And then we do better than most of the existing methods in terms of identifying the binding pocket. So not only that, we use something called a UNet model to go and then segment these pocket shapes. Why is this useful? For the downstream processes, if we exactly know, or if we can map out the pocket shapes well, then we can use that for further processes within drug design, right? Okay, so this is another work that we recently did. Exactly same objective. What we want to identify is the binding residues in that particular in any given protein, but then just based on using the sequence of the protein as the input, right? We do not consider the structural aspects of the protein at all, and then we use the sequence and then try to get the binding pocket. So now one can ask, why is this important? If I can use a protein only if I know the structure of the protein, right? So if I know the structure of the protein, then I can use, for example, the one which I showed in the last slide. So the idea is that if we if we use multiple methods and then kind of connect them in a way where we can improve the accuracy. For example, the last slide I showed an accuracy close to 89%, right? So if I can generate some embeddings from this particular network and then give that also as an input in addition to the structure, possibly we can increase from 89% to a much larger number, right? So so this is another work related to protein structures, though, is that proteins undergo mutations, and we also would like to engineer proteins based on by doing mutations. Like it could be for multiple reasons. It could be for let's say improving the thermostability of proteins in the context of bio engineering or bioindustry, or it could be for increasing, for example, the enzymatic activity and so on. Okay, so what we have done here, again based on the available data of what happens to the stability of the protein with respect to single point mutations. Okay, so we try to come up with an algorithm that will tell us, for any given protein, for any theoretical mutation that we are trying to design, right? So we accurately calculate the difference in the stabilities of these proteins. So not only that, the picture that you see in the right hand side bottom is an integral part of this network, which actually helps us not only predict, right? It's just not a black box. We also try to understand in terms of how the interactions between the wild type residue or the and then the residue that is getting mutated, how do the interactions of that these residues with the other surrounding residues change with respect to the mutation, right? So I will just move to the design part a little bit, right? So Dr. Shastri spoke about like practically unlimited chemical space, right? So the chemical space, or if we just take the number of drug-like molecules that are theoretically possible, it's an extraordinarily large number. There is no way that we can actually explore the whole space and then be able to select what we want. But that's what, in some sense, the objective is, right? What we are in the drug design, for example, or in any other exercise in chemistry, or most exercise in chemistry, is that we want to design molecules or catalysts or materials that suits our needs, right? We want certain properties that has a certain set of certain molecules, a set of molecules that exhibit a given set of properties that we want, right? So let us say this is the whole of the chemical space, and these are the known molecules. For example, in the context of drug design, we look at a zinc the database, for example, is one which is very, very comprehensive. Right? Let us assume the pink one is the zinc database, right? So it has billions of molecules as of today. So now let us say, I want to identify hit molecules from this. Okay. So what does it mean? I have this all the molecules from the zinc data set, and then I want to identify molecules that can bind very well to, let's say, a given target. Okay. And let us assume these are the different molecules that high ranked compounds in whatever ways possible. It could be docking calculations, it could be in experimental binding affinity measurement, it can be anything, right? So based on a certain observation, right, I am selecting top molecules with respect to a certain criteria. And let us assume the crosses that you see on the screen are the ones which we are actually interested in, right? But then this is always not possible to do. Why? Because we are most of the times limited by the budget. It could be computational budget, or it could be a monetary budget, or could be anything. So what we end up doing is to sample a very small subset of the whole space, right? So what that would mean is that if I do that, the ones that crosses in the reds are the only ones I'm going to identify. So what that would mean is that I'm missing out on all the black crosses, right? So what we, one of the things that we have been trying for the past 2 years is to see if we can try to enhance the exploration of the chemical space within the given budget, right? So I'll give, and I will not go into the details of this algorithm, but the idea is this, right? So let us say I have 100 molecules in my data set. Okay, I can do calculations or I can do experiments only about 10 of them. Okay, so but then what can I do to identify all the top compounds? I can traditionally I can only get the top out of whatever the 10 is, right? But then what we want to do though is to get all of the top 10%, right, or top molecules from the whole 100%. Okay, so let us again let's not worry about what the property is and how it is done and so on, right? I can give details if there is a question. The plot that you see is, let us assume that I do the traditional way. I have 100 molecules. I cannot do experiments or computations on all 100 of them. I just choose randomly 6 of them, right? So it turns out that I can only identify the top hits, only with respect to that region. That's what is the red color plot, right? Red color line. Okay, so if I invest 6%, I get 6% of the top hits, right? But what I want is I still want to invest only 6%, but then I want to get 100% returns, or at least as close to 100% as possible. That's exactly what we achieve here by sampling 6% of the data set. Okay, so what we are able to do here is to be able to identify almost 90 to 95+ % of all the top hits that are possible, right? Even if I have done all the calculations or all the experiments, I would still have gotten only that particular subset of molecules. So this kind of gives the overlap of, we call this memes. This particular method called memes, is able to identify a large number of them, even if we consider the whole data set, even though we are sampling only 6%. Right. So this has a lot of applications, especially in the high throughput setting. Again, it doesn't matter whether it's theory or experiments, we should be able to integrate this particular algorithm with respect to anything. So now, as again, the previous speakers spoke about, most of the times we are not interested in one property, right? It's not like, I want a molecule with this binding affinity. That doesn't work. We have to also worry about like the pharmacokinetic properties, solubility, and then the toxicity and all of that, right? So what that would mean is most of the times I'm worried about multi-objectives. So I'm trying to optimize, or I'm trying to identify molecules with respect to a number of properties, right? So we have extended this memes method for multi-objective. Let us say that the blue one is the complete data set, okay? And then the orange ones are like let's say 0.1% of the sample, right? So now what I want to do is I still want to do about 5 to 6% sampling, but I want molecules in the top left quadrant, right? So unless I know all the labels, unless I know the everything, I would not be able to pick. But then I want the method to move, towards picking molecules in the top left quadrant. That's exactly happens. I hope you're able to see the population of orange, which is getting populated on the left top corner, right? So that's the idea of how one can accelerate, or one can make the process of high throughput screening more efficient. Right. So the second, how long do I have, Dr. Panda?
4 minutes.
Okay, I'll try to finish quickly. So the second aspect is, in one of the slides I showed that the chemical space is practically infinite and only a part of it is known, right? What about the possible hit molecules that are outside the known molecule space, right? So how do we explore that region? So far, or in traditional computations, we always talk about molecule to property. I have a molecule, what is its property? But in principle, the question that we are asking is the other way around. So, what the question that we ask is, I want a molecule or a set of molecules that has a certain set of properties, right? So that's the question we always ask, right? So if we can address this question directly, that will be much more efficient, right? Or in other words, what we are saying, instead of going from chemical space to property space, we are trying to go from the property space to the chemical space. So that's basically this idea. I will not go into the details, but then there are methods that we, others have worked on and we are working on, is to see if we can generate new molecules and then try to get the properties of these molecules in a desired region. So for example, the top left one, I want the log P values to be between 0 and 5 for whatever molecules that the algorithm can generate. So that's what you see. Initially, it was in the gray region, and then it has gone to the pink region where it has moved to the center. And similarly, in the bottom right, I want the score to be as less as possible because that's easy for me. That's exactly what happens when I start from the and go. We use a reinforcement algorithm and then see if we can combine physics-based methods and reinforcement algorithm, and then see if we can actually generate, let the algorithm generate molecules that have a set of chemical properties, not just one or a combination of them. So similarly, we use a very popular method called GPT in natural language processing. There are a lot of stories on this particular method in terms of how this particular algorithm is able to actually write pages and pages of text, which is almost as good as an author can write, right? So we exactly use that, except that instead of English, chemistry is our language, right? So in this case, smiles notation, or the characters in the smiles notation, or my letters or words, and then we put together these words and letters to form new sentences, which is equivalent to saying forming new molecules, right? So again, we use this particular novel algorithm to generate new molecules, and we showed that it is actually possible to use this for lead optimization, not just generating random molecules. So I will skip all this, and then I will conclude by saying that there is a lot of potential based on the work that we have been seeing in the last 3-4 years or so, and based on the work that a lot of startup companies are doing, and based on the involvement of the tech giants like Google and Facebook and Intel and all these companies working in this space. There's a lot of potential for making this process more accurate and more efficient, right? So these are some of the things that we have been doing, and then there's some things that we are in the pipeline, and then I will conclude with that, and then just thank the funding and then the students who have worked on all these things. I will stop. Thank you very much. Yeah, thank you.
Yeah. Thank you, Dr. Dugapriya Kumar. I have one question, particularly for all speakers. If with the invent and the highly impact of AI DDR, what will be its societal impact in terms of employment generation? Whether it will shrink the employment, or it is going to increase the employment in the society?
I mean, I can answer, but I think it's best answered by Dr. Shastri or Dr. Subramanian. Can you hear me? Hello.
So, good afternoon, everyone.
Yeah, hi.
So, there are only 4 candidates. Still need time to join the class. The candidates are joining quickly. So, let's give them 2 more minutes.
How? I think there's some crosstalk happening. I request
the administrator can mute everybody so that
Yeah, I was wondering if there is something wrong from my side. So yeah.
Okay. So okay, I think these questions will be put perhaps at the end of this webinar. So now I think we should move to the next speaker.
Yeah, before we move to Professor Sunoj, I just have a quick question for Devapriya Kumar. I was very intrigued with this deep pocket program that you have had. So I was just wondering that can it also calculate the off-target pockets? Because here you're optimizing the binding to a particular target protein, the binding pockets in the target protein. But does the software also gives the likelihood of what kind of off-target pocket would also be available?
I think so. I mean, in the current state, no, but because we identify the binding pockets of the protein that we desire or that we would like to for the ligand to work on, and then since we segment this, the binding pocket shape, it's quite possible that we can explore this shape and then do some kind of an inverse search to on the possible off targets and then see, the compatibility between the two. Yeah, that's an interesting question. It is possible. Yeah, in the current state, no, but that's possible. Yeah.
Okay. Thank you so much. Thank you for giving such a lucid talk. We will now move on to the next speaker, Professor Sunoj. Are you there?
Yes.
Yeah. We've received your slides and we will be able to share those slides and you can just mention when to do next. Should we share your slides?
Yes, please. Go ahead.
Yes. For people who have joined late, I would reintroduce Professor Sunoj. He is a faculty at IIT Bombay and he finished his PhD from ISC Bangalore and he has been a recipient of INSA Young Scientist Medal and Shanti Swarup Bhatnagar Award. His research interest is primarily on quantum mechanical analysis of transition state. And for all those viewers who have put a message in the chat box, there is a YouTube live session on CDRI channel. So these talks are available there on the YouTube live channel. And over to Professor Sunoj.
Okay, thank you. And sorry for the initial mess up a few minutes ago because of slide not loading from my end. And that problem seems to have resolved. At the same time, I think we are entering a really deep neural network because speaker is in Mumbai, listeners are all around the country, slide is in Lucknow. So I think there is an intricate connection that is going on. We hope that the coordination works well so that we can map our X space to the output value Y space. So with that said, let me share what we have been working on in the area of chemical catalysis with focus on asymmetric catalysis. And the previous two speakers have emphasized and given a lot of important remarks and introductory remarks on the power of AI, how it is pervading almost every other branch of be it biology, chemical space, drug discovery, which is the theme of this conference. In every area, you would see the impact of AI reaching too fast, too soon. And there were even questions from Dr. Panda that what would be the societal implication. People have been talking about ethics of AI as much as we heard when nanotechnology was booming about a decade ago. What were the deleterious or side effects of nanotechnology or particularly nanomedicine. So every new wave, this appears like a tsunami. Every new wave when appears in the technological front, whether it is World Wide Web or then the Internet revolution, which ensued, which followed the hypertext transmission protocol, and now it is the AI era, and it is very visible that we have a lot of developments happening. So I would be focusing on a very narrow, I would say, very narrow, admittedly very narrow area of organic synthesis leading to compounds which bear stereogenic centers or axis of chirality, and there are many, many molecules which are in the face of prescription drugs which contain chiral elements. Most often it is stereogenic centers, which would be primarily of carbon origin, carbon with different substituents attached to it, or it could also be heteroatoms such as sulfur, for instance, omeprazole, esomeprazole. These kind of molecules has got chiral sulfur center. So there is a lot of interest in being able to gain precise control over the stereochemical course of the reaction. So when one tries to accomplish precise stereo control, there are a numerous set of delicate factors that might impact. Therefore, empirism is the only way that experimental chemists have been used to, and that I would say will continue to remain so. But the key question is, suppose you are desiring to make R enantiomer of a drug molecule, assuming there is only one stereogenic center with R configuration. And whatever be the process, you will end up making some minor proportion of the undesired enantiomer as well in a typical synthetic chemistry setup. So there is always a question of enantioselective transformation, not necessarily enantiospecific leading to only one enantiomer. So wherever there is a distribution, one should be wary of how much of minor enantiomer or the so-called unwanted enantiomer you can afford to make annually in a multi-kilogram scale production unit. And key questions are, a lot of reactants get wasted in this process of making or letting the unwanted enantiomer form in the reaction mixer. So that is A, it's a waste of money. B, the disposal of such entities is another problem, and you really don't want to get into kinetic resolution or resolving enantiomers, which is like a much harder process, even harder than synthesis at times. So with all this background is hinting at various means to accelerate discovery pipeline in asymmetric catalysis. This is therefore a very important area. Now, what are the tools available? Empirism will still lead the show, as I stated earlier. And there are other methods of quantitative structure activity relationship. One can use very many of these techniques to try to see what are those influential features of the molecular space that would impact your output. That output could be, in this case, in this storytelling, it's about enantioselectivity. So if you could map the X space, that is certain features of the molecule has a perennial impact or a high influence on deciding what would be the Y value. And let me reiterate, the Y value of interest here for this afternoon's lecture is enantioselectivity. So we are looking at enantioselective transformation, where we would have to use a chiral catalyst, what we call as, in asymmetric catalysis, one would try to use a chiral catalyst which forms the source of chirality in the reaction. And a typical reaction would have, let us say, A combines with B to form a compound C, and C turns out to be the one which has a chiral element present in it. And this A and B combination happens in the presence of a chiral catalyst. So obviously, the source of chirality comes from the chiral catalyst. So the questions are, how do these catalysts transfer the chirality to the developing product? So when you look at that, this is a complicated problem, because subtle changes in the substrate, subtle changes in the reaction condition, and even more subtle changes in the catalyst or substitutions which are present in the catalyst can alter the course of this stereochemical transformation. So what you used to have 99.99 enantioselectivity can drop down to something as low as 50% if you were to choose a certain set of substrates or even make a minor alteration in the catalyst, assuming a similar mechanism. That is very important. Assuming a similar mechanism, small changes can make a large amplitude change in the product distribution or the enantiomer selectivity. Why would this change be so delicate, so sensitive? It's simply because these selectivities are imparted at the transition state, and transition state has an exponential relationship with respect to the energy space. So any small change in the energy can have a large impact in the kinetics of the reaction. Therefore, it is not surprising that assuming similar mechanisms, small changes in the substrate or catalyst can lead to very large difference in the outcome of enantiomer selectivity. So this all points to a need for finding the intricate relationships between the structure or the properties. A lot has been said about different properties of interest for drug discovery, be it log P, surface area, and very many other properties which were on fantastic display in last two presentations. So what it also means is that, is it possible that we can make use of non-linear relationship? The linear ones were there in the previous slide. Non-linear relationship between the X space and the Y space, that is enantiomer selectivity, having intricate connection with all the properties or some of the properties of the catalysts or molecules involved. But point to keep in mind is that chemical featureization is a hard task. One can of course extract a lot of these descriptors, molecular descriptors, various kinds of properties by using computational methods. You can set up an automated workflow, collect all the descriptors, but these descriptors are never going to be complete. And if one wishes to do a feature ranking, again,
Dr. Sinoj,
yes?
Actually, you can tell us where to change the slides.
Yes, yeah, it's going good. I'm still in the introductory remark. Whoever is changing is already neural network trained, so it's going quite well, because this is situation driven learning which is happening. So the slide change so far is very good.
We are synchronized with your voice.
Okay, yeah. So yeah, it's good. I will tell in case I want an abrupt change in slide. So far it's going good.
Okay, thank you.
All right, yeah. So we have been looking at this complex relationship between the reactants, catalyst, and the enantiomer selectivity, which I said it's a critical quantity and a delicate quantity. So therefore, linear relationships are not easy unless otherwise you confine your research, confine your reaction of interest to one particular class. Then, of course, multivariate linear regression is a very good method because of interpretability. One can find the coefficients, one can interpret where to change, what to change for a given class of reaction. Unfortunately, the transferability of such methods for a new class of reaction is not easy, and you have to rework the entire workflow to be able to get to this relationship between the descriptor space and the output value. So what I am hinting at is potential existence of nonlinear complex relations between X and Y space. So how do we tackle these? Next slide, please. There are ways of using machine learning to connect. This next slide. Yeah, to connect the physical organic descriptors, which is very commonly used these days for mapping physical organic descriptors to the property of interest, and some of these properties were already on display in previous two lectures. But here the property of interest is a complex reaction space where you're talking about a chemical transformation, a catalytic transformation which has a induction of stereochemistry into the product. So how do we map these? How do we find complex relationship going beyond multivariate linear regression? Just mouse click, please. Yeah, so here comes the onset or inception of potential use of machine learning, and we wondered about this a few years ago, and we have some examples that I would be talking about this afternoon. So the question is, can we capture the complex relationship between features and the output, the features being the ones which are collected from the molecules of interest? In this context, it is reactant A, reactant B, catalyst, and many a times additives, solvent, and other entities which are present in the reaction medium, including concentration of different reactants that are in use. And to see whether we can bypass selecting mechanistically relevant descriptors, that is another question which I will ask myself in the later part of the lecture. Next slide. Okay, so a couple of examples as to lay the foundations. So this is a fascinating example with very rich data set reported by Abigail Doyle from Princeton University on CN cross-coupling reaction. Interestingly, this paper had also attracted a lot of debates, discussions, and it's a very nice study where they have high throughput experimentation done on Buchwald-Hartwig coupling. Those who are in drug discovery would understand the significance of this kind of structural motifs, where you would have a secondary amine through a coupling, a biaryl kind of coupling reaction. So it's a very important reaction. So what they have done is that if you look at the compounds shown in different boxes here, there are coupling partners, a catalyst or ligand, then you have additive in the reaction mixer and different forms of bases. And these put together, and one should think of a concatenation of some kind, but here I'm talking about physical organic descriptors, no technically a concatenation, but you collect descriptors from all these entities, and descriptors, some representative ones are listed here: charge, NMR, molecular volume, surface area, and so on. So these descriptors form the feature space, and these are quantum mechanically derived descriptors. And the output of interest in this particular example is the yield of reaction. Yield of this catalytic transformation is the output value, and X space is decorated with all the quantum mechanically derived molecular descriptors. And here, one point to be noted is that there are certain descriptors, like the third one, it is vibrational frequency and the corresponding vibrational intensity. These are harmonic oscillator approximation vibrations, and here comes a sort of manual judgment. And it would, in my opinion, would be the slowest process in automating this extraction of descriptors. I will come back to this point later on. Mouse click, please. The next example is from the domain of asymmetric catalysis, where one would form a N-acetyl in the presence of chiral phosphoric acid, which is shown in the inset with the dotted lines. So chiral phosphoric acid, the range of chiral phosphoric acids have been used for this transformation, again of relevance to biologically active molecules. They have introduced a different sort of molecular descriptor, what is known as average steric occupancy, somewhat complex description, but it would capture the spatial spread of this catalyst, particularly when you look at the R substituents at the 3' positions of the binaphthyl phosphoric acid. The R space, how does it span, how the rotations, how the conformation of these R substituents would impact the enantioselectivity of the reaction is essentially the theme that is being mapped here, that depending on the catalyst structure, how would the enantioselectivity vary, and that is on a closed set of phosphoric acid catalyst reactions where the sample size is about 1,000 plus. Next slide, please. So with that said, let me move to what we have been trying to accomplish. As I made in the half introduction before Deva's talk, I said that we wish to work in the small data regime because that is where the developmental chemistry take place. And big data regime also means that chemistry has evolved quite well, maybe over the years, maybe through high throughput experimentation. We do not wish to get into high throughput or big data, because we want to contribute to an ongoing development, ongoing synthesis, ongoing asymmetric catalysis problem. So what we have done to demonstrate a proof of concept to begin with is to look at asymmetric hydrogenation, which is again an important process. Down in the third row, you will see a alkene and imine. Those are the substrates, and hydrogen in high pressure is the other reactant. So this substrate gets hydrogenated in presence of transition metal catalyst L1 to L4, or you could accomplish the same thing with L5, which is an organocatalyst, operates through an entirely different mechanism. And so these 5 catalyst families are used as the source of chirality. And now, how many samples do we have? And I must also acknowledge that these samples were manually curated from publicly available literature. No major database is used here, manually curated. to collect the asymmetric hydrogenation reactions spanning about decade plus of research results spread over several papers. So this forms our data set, which consists of 368 reactions, of which there are 96 of alkenes, 94 imines, and different catalysts are shown in parentheses. There are about 58 unique catalysts. Next slide, please. So this is the diversity. Sorry for having this very small, tiny postage stamp sized pictures here, but I just want to emphasize on the fact that there are quite a bit of diversity. If you look at the last box on the lower half, you see the diversity of alkenes and imines. There are a huge number of structural diversity, linear, cyclic, aryl, biaryl, all sorts of diversity in the alkene and imine part. Those are the substrates which undergo hydrogenation. And then we have diversity on the ligand family. What are the substituents on the 3,3' positions of the ligand? Those are listed in the top row for different ligand families. Next slide. Here comes the very critical aspect of featureization, because we wanted to develop a unified model. So we were very careful in identifying features that are to be selected. Of course, there is a mechanistic backdrop in this. How do these catalysts operate in a typical mechanistic scenario? What are the important steps? How do they bind to the transition metal and so on? All these are considered. So we selected molecular descriptors such as bond distances, NMR chemical shift, vibrational frequency, charge on various atoms from the highlighted regions of these catalysts. These are common such that our data matrix remains symmetric across samples versus features, and then the same data matrix will include that features collected from the alkene or imine subunit from the substrate, and local descriptors as well as global descriptors were collected, and that forms our X space, and we are training different machine learning algorithms to be able to find the relationship between the X and the output value, which is enantiomer selectivities for these reactions catalyzed by this different family of catalysts. Next slide. So this is how it is. There is a feature extraction step, which could be regarded as slow depending on what kind of features you want to extract. I said earlier that vibrational frequency and intensity would require manual judgment whether this vibration belongs to the inter-binaphthyl vibration, because that is critical, or many of these requires bit of manual judgment. And there are, of course, polar surface area, ovality, many other global features that all got captured into this input matrix. And we have for each sample several features that constitute a given sample and feature, and there are 101 features, 368 reactions which are mapped onto the Y space, that is enantiomer selectivity. Next slide. What did we find? So this is the summary of the key results. So if you look at the L1 family of catalysts, that is phosphoramidate family used for asymmetric hydrogenation, we could get a root mean square error performance of 6.3 with a standard deviation. So 6.3 would roughly mean, if you want to convert this into a number equivalent representation, 6.3 is good vis-a-vis the enantiomer selective transformation, which is reported in literature for other couple of reactions or yield predictions. They are roughly around 7-ish. So 6.3 is a decent estimate of decent error that one can afford to get. Now, we did not stop there. We went on to infuse one catalyst family with the other, and so that if you look at the last row of this colorful diagram at the end, we see that all catalyst family put together to form a unified random forest model, which returns a performance of 8.4 root mean square error. And how does it translate into numbers? About if you make 100 predictions, 90 to 92 of them are within about 10 units from the actual experimental enantiomer selectivity. So that is quite good given the complexity of this reaction space. And this is also a pictorial representation. If you look at this green mat, it's mostly green with a tiny little red dots here and there. So if you mouse click, an expanded version of one of those runs, and these RMSEs are, please mouse click, please. These root mean square errors are collected over 100 independent runs, which will have a 80:20 test train split, so that sufficient randomization is ensured. We are reporting average root mean square error over 100 independent runs. So this is an expanded version to show. I do not know, is there a setting problem? I keep getting requests for admitting new participants. Is that okay, or because it comes in the middle of my slide?
There is no problem, sir. I am admitting it from here.
Yeah, but it is blocking my slide actually. So
no,
right now I cannot see about 20% of my slide because someone wants to join.
Okay. Actually, actually there is one participants option. Just click on there. Actually, then you will not see this thing.
Oh, okay.
There is a participant option. Okay.
Yeah. All right. Yeah. So now, if you look at the green patch on a typical run, it has about 3-4 values are terribly predicted, but by and large we can live with such a predictive power of around 10 or low. Therefore, that is the kind of result that we have. So next slide. Always the question is the transferability of any machine learning model. These are small data set problems. Here, what we have tried, and we had primarily trained our network using binaftyl system, which is like two naphthyl rings connected, and there is a whole bunch of phosphoramidate, phosphoric acid, and so on. So here, these are purely out-of-bag samples, which are of biaryl origin, and we predicted for them, and it turns out that the root mean square error for this purely unseen reaction class is about 8.5, comparable to that of the unified model, which is 8.4. So this again ensures that the model, random forest model that we have developed, among many other methods, is transferable enough for asymmetric hydrogenation within the territory of asymmetric hydrogenation, irrespective of the catalyst structure. So that was quite fascinating. And next we move on to, next slide. Move on to because I have repeatedly mentioned there are manual interventions required for the descriptor extraction. Feature extraction can be a slow process. So we wondered whether we could resort to alternative ways of representation learning and make use of available data in other related domains. So that takes us to a situation where we may be able to reduce the computational cost and error in judgment when there is a human intervention. So that is what we have tried to do in the next process. Mouse click, please. So here, given a catalyst or a set of substrate, or substrate and catalyst put together, can we use some sort of a learned method? So we first transform these chemical space into string-based representation. Deva was talking about SMILES, and the previous speaker, Professor Subramaniam, also talked about SMILES representation. This is one of the popularly used linearization of molecules, so we have a string-based representation. So we try to map this string with the output, and output again is enantiose selectivity or yield, which is in the complex reaction space. Next slide, please. These are the examples which are selected because of multiple reasons. One, there is benchmark available. These reactions are known using molecular descriptor-based approach whose performance can be compared. And second aspect is about data distribution. The first one is quite rich, very dense and uniformly distributed data. These are output values. Yield is quite uniformly present in all regions for across samples. Now, reaction 2 is the Scott Denmark's example, where you see again data distribution is quite good. You have samples in all ranges of low E to all the way to 99.99. Again, it's a good example to study. But the third one, that is a typical practical scenario when you're talking about reaction development, when you're talking about asymmetric catalysis development, where there is sample scarcity in the lower data regime and reasonably dense in the higher data regime. So this is a classic case of class imbalance, and we wish to deal with them using both descriptor-based learning and representation-based learning using molecular string. Next slide, please. So I think it went too fast. Of course, I think for this audience it may not matter because this was meant mostly a pedagogic reason. How do we combine all the entities in the reaction medium So we have a ligand, which is a chiral ligand, catalyst, and then coupling partners and a base. All these are concatenated. That forms a sample. Next, mouse click. And those are tokenized, numericalized. Next, converted into one-hot encoding. That forms our vector space. Next, click. And this forms the input to a natural language processing model. We have used ULM-FIT for this. And one can quickly grasp the network architecture. We have an encoding layer, and then 3 LSTM, independent LSTM, fully connected LSTM layers. And then we have linear, softmax, and then map on to the regression problem. So we have this enantiomer selectivity or yield. This is on the network side. We use a recurrent neural network for this problem. Next slide. So what did we find and how we made use of this transfer learning concept? So we have this ChemBL database consisting of 1 million molecules, unlabeled data. So we wanted to use them and pre-train our language model. ULM-FIT was used for pre-training with this 1 million molecules represented in the form of smile strings. And we have fine-tuned it for a regression problem, and then applied to yield or E prediction on examples which I have shown before. Mouse click, please. And then we want to transfer the weights and bias from what we learned from the 1 million pre-training exercise, applied to a target-specific task, which could be Buchwald-Hartwig coupling, N-acetyl, asymmetric N-acetyl formation, or asymmetric hydrogenation. So this transfer learning is beneficial provided the learning which occurred in the first phase of the work is beneficial to the downstream task, and we can customize for a given reaction. So that is what we were trying to accomplish in this work. Mouse click, please. This is the summary of performance. As you can see, reaction 1, which is dense in data, uniformly distributed. Here TLM1 is a transfer learning model directly by using the ChemBL molecular assembly or molecular library, where we don't do any target specific fine tuning. So we were able to collect a root mean square error of 6.8, which is good. And if we were to fine tune on the data, sorry, I think I just flipped. If you use the direct method, it is about 6. And if I endeavor to fine tune using TLM2, that is fine tune on the target specific, that is this Buchwald-Hartwig amination data, then we have a slight erosion in predictive power, but still 6.8 is quite good. Now we go to reaction 3, the top chart that you could focus on. Then we see that the transfer learning is transfer learning that is in orange color, that is a superior predictive power, that accomplishes a superior predictive power compared to the one which uses the direct use of the ChemBL molecules. So that means, depending on the problem at hand, one may be able to make better use of transfer learning. There you're completely bypassing the feature extraction through the quantum mechanical workflow, and you can directly get access to the predictive power. So you have this trained model, and one can use this for out-of-bag predictions for all cases of Buchwald-Hartwig amination or N-acetyl formation or asymmetric hydrogenation. So we have this robust model good for predicting reaction outcome, yield, or selectivity. Next slide. So if one wants to look at in terms of numbers and the quality of predictions, if you focus on the overall quality across all the reactions, about 91, 70 plus 21, 91% of predictions are within about 10 units from the actual value, which is actual value means experimental value versus what predicted by the NLP model. So that's about 91%. Now, if you look at specifically yield versus enantiomer selectivity predictions, if you look at the, yes.
You have 4 more minutes.
How many minutes?
4.
Yeah, that would be perfect. Yeah, I'm almost there at the end. So if you look at the lower leftmost pie diagram, that is about 91% of predictions are within 10 units. That is for the Buchwald-Hartwig amination project, and it is also a subtle reflection of density of data and number of data points. This is about 4,000 plus uniformly distributed data. The transfer learning protocols are helping us gain predictive power within about 10 units. 93% of predictions are of that quality. When you look at the initial selectivity with differing data distribution and differing data dimension, one would see that even there, 88% for N-acetyl reaction and 90% for asymmetric hydrogenation has about 10 units from the actual value. So these are all exciting times and days of customizing AI for practically relevant chemical reaction space. This is what we have tried to do. And next slide, just to summarize that one can bypass the feature extraction and make the process more time economic by using molecular string-based representation and use a rather complicated natural language processing network architecture and convert them into a regression problem. And that was, we were able to get around, on average, 90% predictive efficiency of samples, including unseen samples for prediction of catalytic activity. Next slide. So this is the summary of descriptor-based approach using molecular descriptors, quantum mechanically derived on the left column, and the merits of the string-based approach or the representation learning method using NLP kind of network. We have a summary of good points to make, that comparable predictive accuracy. So this is one of those examples where direct benchmark comparison has been done between a natural language processing architecture and that of a random forest model built on quantum mechanically derived descriptor space. So we are trying to see how effectively this transfer learning can be plugged into active learning, where one would also route it through an experimental pipeline. I have some collaborators who would be willing to test these experimentally, because what I have not talked about is generative models, which Deva has been referring to in his talk. So we are working on that space as well to see whether we can trigger an active learning process in the area of catalysis. Next slide. I must thank my graduate student Sukriti Singh, and in fact all slides are the slides which she has used in a recent conference talk, and I should thank her for her enthusiasm in working on these rather complicated projects in the machine learning in asymmetric catalysis. And some of our good friends here in IIT Bombay, Professor Balamurugan, who has been quite helpful in many discussions, and Preeti Jodi, who is a expert in speech recognition using AI. And of course, a lot of these computations could have been possible without the help of IITB supercomputer. And I thank all the members of my group for enthusiastically participating in AML workflow in the group. Thank you all for listening.
Yeah, thanks a lot for presenting a very different topic on asymmetric catalysis. If I may ask this question, maybe it's a naive question, that sometimes both the enantiomers have activity at certain. They show activity at a different percentage. For example, 70-30. So can you predict what should be the catalysis rate so that we get that population of 70-30, or is it possible?
Right. And technically it is possible, but there is a cultural clash here when you think from an organic chemist perspective, that they are super happy with more than 90% EE, and 99 they will be jumping off the floor. But 70% nobody is happy, which is a reality. So if you want to induce academicians to get interested in a distribution of 70-30, one should have a specialized program to convince them that, look, we are interested in such a mixture without going into resolution problem, because that will make the things complicated. Yeah, so it is a matter of culture that is why in asymmetric hydrogenation we don't have many samples in the low E regime, because culturally they don't report it, right? And it is not good for a data science perspective that if you have well distributed samples learning would be far more effective. But I think it's a matter more to do with the culture of organic chemistry.
So, so what kind of experimental models are required to make such kind of pipelines?
One can go in the slow track, manual, empirical experiments, or one can also set up robotic stations designed for carrying out certain types of experiments. It may not be versatile enough. Asymmetric hydrogenation would be hard, but a single experimental setup can have a turnaround time and one can do it. But if the other reactions like Buchwald Hartwig was entirely done on high throughput experimentation setup. So those are, like, of course, expensive in terms of instrumentation, but it is affordable if you were to customize your reaction and develop your own automation system.
Okay, so there's another question from a viewer. So he says that other than C2 symmetric chiral catalyst, any C1 symmetric CPA molecules have been analyzed in asymmetric hydrogenation reactions?
Analyzed as in if it is the AI based analysis, no, maybe there are some samples. Actually, if I go back to this slide where I have the structural diversity of catalyst, I'm sure there would be some C1 symmetry catalyst. Yes, it is part of the data set, but we did not specifically look at a C1 case. But it should be all inclusive. It wouldn't matter.
Okay, so you've done this AI based in your study. This is in one of your slide. You're saying that you have looked into such kind of data set.
Yeah, there were samples which are C1 symmetric.
Okay.
So one should carefully look at the sample diversity of catalyst, and there are like R1 not equal to R2 kind of situation. So naturally, it is not C2 symmetric.
Okay. With this, I would like to thank you, Professor Sunoj, and it was a very interesting talk. And now we move on to the last speaker, that is Professor Ahmed. Professor Ahmed, you're there, right? Okay. So Professor Ahmed is working at School of Computational and Integrative Sciences in JNU. His research interests are machine learning and big data analysis, and his lab primarily uses a broad range of biological data such as DNA shape-based transcriptional regulation, single-cell transcriptomics, and using data from clinical data for medical diagnosis. And especially for the viewers and the students, I would like to mention that he has a very interesting website. Please go through the webpage of SciY of his lab, and you will feel very motivated after reading some of the things he's mentioned on his website, especially when he writes that his lab believes in creativity, rigor, excellence, and strive to improve ourselves. And I think as a researcher, it's important for all of us to have that kind of temperament. And with this, over to Professor Ahmed. He would be talking on AI and data-driven techniques for genomics and system-level drug discovery. So will you be sharing from your end? Professor Ahmed, I think you're muted. So are you able to? He's able to.
Can you see my screen? No.
Yes, we can.
Okay, but just one. So you see my first slide, and let me go full screen, and then I have to check whether my slides are changing, because that is sometimes a problem. Are they changing?
It's not full screen yet. Not yet. Not yet.
That's why I think so. I'll share the full desktop in that case.
No, sir, it is okay, sir. Just click on full screen. I think then.
No, when I do full screen, usually it disappears. That's the problem with Microsoft Teams. Many people face. So I'll share the entire screen. Then I think it will be best.
Oh yes. Actually, I think probably you have shared only presentation here.
Yeah. So I think it works now.
Yes, it is working. Yes.
Okay, great. So thank you very much. And thank you for paying attention to my website and to going through this. It's so nice to connect to CSIR family. I'm kind of a member of the extended family, a relative, working in this field. So today I'll be talking about artificial intelligence, and based on being the last speaker, there are some disadvantages because certain things you want to say have already been said. But there is also an advantage that you can draw upon and you kind of extend the argument or learn from what the speakers have said and try to put your view on this. So it gave me this is my lab. I can skip it because we don't have so much time. I start with a poem and then I'll make a remark about based on what has been discussed, and based on that I have understood the audience better. So with this little poem, that physics is fundamentalist and fussy. Mathematics is dumb and not easy. Astrology is hasty and crazy. Why I say this is physics based on fundamental laws, and it requires a lot of rigor. Mathematics, why I say dumb, apologies to the mathematicians, is dumb because it requires predefined equations and tries to fit the data into them. And astrology is too hasty and crazy because it can draw conclusions from very few observations, which may not hold in new scenarios. But human brain, actually, when we do our acts in our lifetime experiences, when we interact, when we take decisions, when we learn, we do not use either rigorous mathematics or deep physics or astrology. Our brain, because for deep inside its terrain, it knows to represent, test, and train without bothering to explain. And this is the essence of the intelligence. Data science rules this world, for even if a solution is transient and predictions are noisy and incoherent, they work the best for most because they are intelligent. So now, since the discourse was about AI, ML, and data-driven knowledge, so this often we get little overlapping ideas about what is artificial intelligence, what is machine learning, and what is data-driven knowledge. So I put a few points across just for the speakers. Just a moment. I think my full screen is not visible. I have YouTube running in the parallel, and I can only see the not the full screen.
No, your screen is visible. We can see the AI ML data-driven knowledge slide.
But YouTube sharing is probably of a different window, is it?
I think the YouTube, but there is a delay. There's a delay of a few slides.
Oh, okay, okay, that's right. So I had YouTube also streaming in front of me, so I was wondering if it is correct.
No, there is usually a gap, a delay.
Yeah, I got it.
Okay.
Thank you. Yes, thanks. So first of all, intelligence and learning are often confused by even the people who are working in the field of artificial intelligence. Intelligence as such is not about learning. And if you talk to cognitive scientists, they will say that intelligence cannot be trained. But yes, intelligence is the ability to learn. Somebody who is more intelligent, they can learn things faster. But as such, through learning, through training, you cannot make somebody intelligent. Cognitive scientists will tell you that you cannot modify the intelligence of a student by just putting him in a classroom. So essentially, what we do through learning is trying to make people or machine to behave like intelligent. So intelligent solutions come from internal knowledge representation and the ability of solving problems. Also, in the context of data science, it is very important, and it is important in general in our life also. Trained is not intelligent. Overtrained can be sillier than the untrained. And this is one of the most common problem in data science or data science issues when we solve any biological problem or any social science problem or any chemistry problem with the help of machine learning. We often overtrain our systems. And greater challenge today is not about training our models or creating the trained models. Greater challenge today is about avoiding the overtraining, and so that the models, the solutions that we acquire through this data become applicable in scenarios in which they have not been trained. So overtrained, therefore, is sillier than the untrained. And as I said, learning is a process of copying the intelligent. There are many in artificial intelligence also. There is a topic of artificial intelligence where we kind of learn, for example, what is the fastest way of reaching a state, agent state, from one state to another. What is the fastest way of going in a graph from one point to the other? So these kind of solutions can be obtained with the help of mimicking how a human brain or how the intelligent systems, natural living intelligent systems, such as the communities, such as the ants, such as the societies, have developed a kind of intelligent method to find solutions for those problems. And we get inspired by those solutions, and we, instead of solving these problems exactly through the mathematics or through deep equations, we try to learn that we can perhaps arrive at these solutions faster, because equations that we use today, any equations of motion or equation of forces, they have not been designed to solve bulk problems. They have been designed to solve particle problems. When we do in physics, the first principles of equations of motions are designed for point objects. But when we try to use them for bulk systems, converting these ideas of point objects into a bulk system becomes complicated. And that is how the human brain actually learns to work from the systems level. It does not look for the individual entities. Child learns a language, child learns grammar without having to know exactly what is subject, what is verb, what is object. So through the process of learning, the data is converted into internal representations, which is not atomistic in nature. And therefore, this kind of learning approaches are inductive in nature, whereas intelligence may work either in an inductive manner or in a deductive manner. We may never have seen a data. We may have come across a completely new situation, and an intelligent person can solve this problem better than somebody who has been well trained. So that is where the intelligence just is different from learning. Finally, data can be modeled either with the help of a machine learning. You have a lot of data. You represent it as a kind of inputs and outputs, and then you try to develop a machine learning model. It can be a neural network, it can be a regression model. Based on the complexity that you require, you choose a model and you try to find a relationship. You do not see that relationship. You may or may not require interpretation of that model, but you want something, a black box or a white box or a transparent box, which can relate the set of inputs that you have and the set of outputs that you desire from them, so that after the training, the system becomes ready, that when you have new set of data for which you do not know the outputs, it will be able to give you the answers without having to bother about why it gave an answer which it gave. So data does not necessarily have to be modeled in this way. Data can also be modeled in exact mathematics. You can define equations, and then you can try to use least square methods, or you can do simply curve fitting. So this is not really a data-driven approach. This is also a data-driven approach, but it's not really a machine learning approach. So both the approaches are good enough for the data science. So that is where we distinguish between how we use data. We use data either through machine learning or we use through mathematics. We'll do learning, and learning can make systems to behave like they are intelligent, and intelligence itself works in a manner to solve problems with or without machine learning. So now coming to the question of drug discovery or drug. So this kind of slides must be cliche for you. All of you are drug experts, and you must have seen these kind of slides plenty of times. But I just wanted to draw this attention that there is an attrition rate in the drug development, and the basic science, basic research produces 10,000 to 30,000 substances, which result into one product in the phase 4, and it takes about 12 years to go from this stage of basic research to the final product. So where is the problem? Well, basic science is not doing a great job. Basic science is producing too many candidates, out of which very few are getting selected. That's why I put the remark on the right that the basic science needs to get smarter. There is a need for the basic science to become intelligent. Instead of producing 30,000 candidates in the first stage, it should produce at least, at the most, 1,000 candidates, so that the cycle and the attrition rate can be reduced. And the reasons for rejection have been reviewed by many. I mean, maybe you people have much more knowledge than me in this field because you are drug discovery experts. The efficacy and safety are the primary reasons that these drugs have been rejected. So can we, at an early stage, look at these drugs and look at their systems level response and systems level effect, and tell about the efficacy and safety right in the beginning when we are generating candidates? So that is where there is a need. So in terms of the drug discovery process, the basic science can be benefited in the aspects of understanding the molecular basis of drug and therapy. Then you identify targets, as one of the speakers said. When we have a target, this target may not be druggable. But so there is a question on looking at the proteins, looking at the DNA genomic targets. Can we tell whether these targets will be druggable or not? What is the systems level impact of a drug? So can we, a priori, instead, when we do a docking experiment, when you do in silico drug screening. So at that time, we generally do not look at the systems level impact of the drug. What are the advances that have been made to look at the drug and tell in advance that this is the kind of transcriptomic or metabolomic changes that can be brought by this drug? And in this way, if we are able to know and predict something about the systems level impact that this drug can cause, we will be able to reduce this number of 30,000 to a smaller number. Then same way, the AI and ML applications can help us in determining the effects, the side effects. The side effects come from the systems level study of the drug impact and intelligent repurposing. All these topics already covered in the previous talks in a very nice way and in a very much more professional way. So that is how the AI and ML. AI/ML can also help us in diagnosing diagnostics and treatment selection. So if you are doing it is not just about the choice of a drug, but also the dosimetry, about how long you give a particular drug, etc. So in the treatment selection, when you also personalize the medicine, so the personalization in that case can be done with the help of profiling a patient, profiling the disease condition, and developing diagnosis. So this profiling is a process of diagnosis itself, and also diagnosing early stage discovery of a disease or telling what is the a patient suffering from. So in our lab, we have a number of projects running, which may be, I showed you that we have a big lab. So a lot of projects and different kind of projects, complementary and sometimes totally independent projects are carried out. I'll share with you some of the experiences that we have had of using AI/ML or data-driven biology. So I put these three together, but I already clarified what is the relationship of AI/ML and data-driven science or biology. First of all, I'll talk about the biomolecular interactions and computational structural genomics and how we have applied AI and machine learning models to solve or to understand biomolecular interactions. In the second part, I'll talk about the drug prioritization with the help of transcriptomic data analysis. Third part, I may skip if I do not have time, but at least I'll be able to cover the two. Third one, I wanted to talk about integrating the sequence context and the transcriptomic context so that we can talk about functional notations in a specific cellular state. So first work, when we have been working on biomolecular interactions, we have worked on a number of different types of molecular interactions. Most of frequently and with the greatest passion, we have pursued DNA binding proteins. Why DNA binding proteins? Because DNA binding is one of the most important biological phenomena. It starts with transcription. So all the genes are expressed through a process of protein interaction with the DNA, genomic DNA. But not just that, DNA of the pathogens is recognized by certain molecules, the DNA nucleic acid sensors, which tell that there is an invasion, and they activate the whole process of host defense. So therefore, DNA binding proteins are very important class of proteins. And one of the recent relatively studied we studied was the conformational changes, because very often we talk about the structure-function relationship. A lot of biology, a lot of structure-based drug design is based on a relationship between the structure and the functional implications. And we are all intrigued, and we are also all troubled by the fact that when we look at the protein in isolation, and when we look at the protein in complex with its ligand, or its DNA is the ligand in this case, there is a lot of conformational change that this protein undergoes. And therefore, any rigid docking or rigid understanding of the protein DNA complex or protein ligand complex leads to a lot of problems and does not give us the right answer. So we characterized what kind of the conformational changes happen in the DNA binding proteins, and we identified there are 6 types of conformational changes, and each of them has a different character in terms of what kind of free energy change will happen when the complex is formed, what kind of specificity will be observed when a particular conformation changes. So for instance, if the protein undergoes a large conformational change upon DNA recognition, it is highly specific, and it is the most stable also. So many such things emerge from this analysis. So one of the questions is also in the genome, there are a lot of proteins for which we do not know what kind of function they do. So we, instead of trying to do a general notation and trying to do a homology sequence similarity based approach, we also look at evolutionary profiles and just trying to predict a specific function of DNA binding proteins. So there are many proteins still in the proteome, for which they may be potentially acting as transcription factors, or they may be acting as another kind of protein DNA interaction. So where we use the deep learning and we try to represent the evolutionary profiles, because any protein that is placed in under evolutionary pressure will evolve in a particular way if it has a given function. So a residue will, or everybody knows that if a residue is conserved, is functionally important, it is more conserved. But just being conserved is a very little information. But a residue undergoes specific changes if it has a specific function. So for instance, arginine just getting replaced by any other residue is not important, but arginine getting replaced by lysine and arginine getting replaced by alanine will tell us about what kind of arginine is this. If arginine is frequently getting replaced only by histidine or say lysine, then it is most likely there is a role for the positive charge. If arginine in the profile is getting frequently changed to alanine or to say aspartic acid, etc., so probably charge is not playing any role in this evolutionary profile, and therefore we can say it may have another function, not the function of the DNA binding. So these kind of behavior properties can be enumerated if you look at the physics and science of it, or we can just put everything into the model in a CNN or in a multiple linear regression model and try to let the model find what is important and what is not important. So that is where machine learning plays a role. In either you identify, you characterize the features which are important for you a priori based on your knowledge of physics, chemistry, or any domain specific knowledge, or you just put everything that you know about the system, put it into the model, and if there is a lot of data, then automatically the model will filter out, okay, this particular property that you're looking for is not dependent of feature XYZ, and it is dependent on feature RWT, etc., etc. So let the model discover. So this is the kind of approach that is taken in machine learning. We do not bother about what features are giving us a next answer. What we bother about, the answer should be correct. So second question is, as I was saying, that we have been talking about the AlphaFold. So predicting the structure of a protein has become much more accessible today. And if you have a protein sequence, you can possibly tell what kind of fold it will undertake. But the whole trioca of the sequence, structure, and function is not really complete, because sequence leads to a structure. Structure is much more robust. Within the same structure, if a single residue change happens, then the entire interaction changes. So it is also important within the same structure, what is the role of the sequence? So sequence-based properties are important, and therefore we have worked extensively on finding the amino acid residues which actually take part in binding to DNA. And some of them were based on, say, the clustering of the conserved residues, that is the amino acid residues which form three-dimensional clusters and come together. They play a greater role in the stability of protein DNA complexes, whereas there are some conserved residues which live alone, and the lonely conserved residues do not play a much bigger role, much role in the formation of protein DNA complex. So we therefore identify the nature of conserved clusters that happen around the conserved residues or with the participation of conserved residues, and what kind of changes they bring about. More detail, if we look at the sequence level, so we can say, since DNA is negatively charged, therefore positively charged residues must be contributing to the DNA binding. Therefore, we can say all the arginine must be binding, but all arginine are not binding. So the sequence level neighborhood is important. So how we incorporate into the secondary in the sequence neighbors. The moment you go from one residue, which is 20-dimensional information, if you want to go to 2 residue, it becomes 400-dimensional information. You go to 3 residues, it becomes 8,000-dimensional information. So this high dimensionality that arises because of the increasing number of the window, the increasing size of the window that you are considering, this cannot be modeled by traditional mathematical expressions, because the number of parameters you have to train now become too large, and you do not have so much of data to find these parameters of a mathematical equation if reliably. So what we do is we represent the entire sequence as positional information, and if this position is X is occupied by arginine, if second position is occupied by whatever residue, we give a sparse representation, and we then create a much reduced representation of a window and then utilize the machine learning, because now we cannot create a mathematical formula to do this kind of representation. So neural networks works best. So we started solving this kind of binding site problem on the sequence and the structural properties way back in 2004. And we did prediction earlier for the solvent accessibility of amino acid residue from sequence. So that work I'm not showing here. So amino acid secondary structure, solvent accessibility, DNA binding site, etc., they all can be modeled in a kind of similar manner, you have a sliding window, and within each window, you have a reduced representation. You represent it by an evolutionary profile, and you represent it by sparse encoding, and try to develop a machine learning model. So other models also we developed earlier. That one was carbohydrate binding site prediction way back in 2007. And some of these models we are still working and trying to improve, but some of them we are not working anymore, but just for a historical perspective. So these are the kind of different sequence-based properties on proteins that we have investigated in the past. So another very important activity that we did recently was to incorporate AI and ML to do intrinsic versus context-specific information in molecular information. I'll explain what I mean by this. So first of all, this is the work we did earlier. Again, one of the speakers talked about the knowledge-based force field. So this is a data-driven approach. So this is where you would know that this is a data-driven approach, but this is not a machine learning approach. In the data-driven approach, we take the protein DNA complexes from the protein data bank, and all these structures are taken into account, and we treat this interaction between protein and DNA as a kind of a force field, and we create what we call the knowledge-based force field or statistical force field. And in this way, if we modeled what we call the technique called readout, and it will tell us whether the given protein DNA complex, what kind of a specificity it has with DNA sequences, and whether the same protein will also bind to other DNA sequences, and which are the sequences which can bind more strongly compared to this, or which are the sequences it will not bind. It has a very successful impact, and we did some experimental collaborations, and many of our collaborators, some of them, at least one of those studies was published, where we found that there was not a canonical DNA motif, and actually the protein was binding there. And it was binding because the DNA was acquiring a particular structure, and that structure could be modeled with the help of this statistical force field. So we did these predictions, and we got them experimentally verified. So now, if we look at the transcription factors, again, since we're talking of protein DNA interactions, in different cell types, can you just alert me about the time so I can? How much time I have?
Dr. Ahmed, you have 5 more minutes.
5 more minutes, okay. So just quickly, I think I'll skip this one. I would like to introduce another resource. I'll just make one statement about this. So in this, using the AI model, we have been able to do a whole genome analysis of molecular dynamics ensembles. So what we have done is we created the conformational ensembles of all the tetramers and then mapped them to an entire genome. And we created the dictionary that if this is the sequence, this is the likely to be the conformational properties, dynamic properties of the sequence at this level. So this allowed us, because instead of doing a real-time simulation, creating a dictionary of these simulations, we were able to analyze the entire genome. And we found that this model works very well in predicting the transcription factor binding sites. So this was a success story which we published, and we also predicted the modular regions of the transcription factor binding sites around the motifs of these STAT3 is the one example shown on the screen, but also for other transcription factors. So one particular project that is of interest for the drug discovery is the CMAP project, which has been developed by a consortium, Connectivity Map. So what has been done in this project is that the idea is that if you have a particular disease and you know in this disease, this is the transcriptomic changes that takes place. So if you have a priori collection on different cell lines, if you treat these cell lines with a particular drug and you look at what is the expression level change that this drug is likely to bring about, then you can look at the disease transcriptomic change, and you can look at the library of the drugs that you have previously compiled, and you can see which drug is likely to bring about the change which is just the opposite of what this disease is causing. So in this way, instead of doing a structure-based or ligand-based approach, you are just looking at the systems level approach. System here is just the transcriptome data. So you can do a mapping, and a new drug can be, a priority candidate can be selected, which is likely to bring about the changes, not in just the gene of interest, the gene of target, but the entire systems level. So this database, which is already available, not our data, but this comes from Connectivity Map data. This has data from 15 cell types. 5,000 compounds have already been deposited there in the public domain. And for 3,700 genes, they have this data. So this is the work we try to extend, and we try to do that in the data, because the data is limited. You do not have too many drugs there, and you do not have too many disease conditions there. So we try to expand this to by taking the gene expression data from Gene Expression Omnibus, which is any experiment on gene expression that has been done in the past has been deposited there. One challenge there is that this people have done experiments on expression in microarray chips. Somebody has done on GL570, somebody has used GPL596, somebody has done RNA-seq, and some of them have 20,000 genes, some have 15,000 genes, and the expression data is, in some cases, it is the level is 0 to 10, in some cases it is 0 to 1 million. So all these kind of scaling problems, data normalization problems, missing values. So all this data, when somebody tries to do a meta-analysis, combines the data from a disease, say let us say somebody wants to look at the lung cancer, and they can extract the data from lung cancer from GEO. But when they look at, there are 100 studies on lung cancer, but each of these 100 studies is giving you a different quantification of the expression levels. So putting them together, it takes a lot of time. So what we have developed is, we have developed a huge expression data matrix, which covers 5 platforms and 54,000 probes, which is roughly equivalent to 20,000 genes. And then wherever the gene and the platform chip was smaller, in some case, for example, in the case of GPL570, you have data coming from only 978 landmark, 11,000 CMAP target genes, but we added the expression values for 9,000 genes by computationally predicting them. We added in this platform 756 genes, which were not available in the expression platforms. And how we did it? By pulling together all the 5 platforms, and whichever gene is missing, we estimated this with the help of a deep learning model. And with the help of deep learning, we could reconstruct the missing values. So we have now a complete platform which has 20,000 odd genes, and this data is now available. This paper is under review. So this MPGEM, so-called data multi-platform gene expression matrix, will save a lot of energy. If you want to analyze some disease condition, some gene expression condition, under cellular drug condition, or there's so much of clinical data there, including toxicity data, etc. So you can search for those keywords, identify the samples that you want to investigate, and collect the data from MPGEM. And in a single attempt, you can go to the next level of identifying what are the marker genes representing this condition. So third part I'll skip because I think I've already run my time. I'll just quickly come to the summary. So systems level discovery of novel targets requires a look at sequence structure and gene expression patterns in a biological context. A novel target discovery based on interaction predictions have come of an age and can be incorporated into the lead generation effectively. Reverse transcriptome analysis is what I presented in the last, provides an alternative and complementary structure-based drug design approaches. Finally, I have a small poem again for the AI and ML problem. If you remove R, B, and L, it becomes a poem. So it says, mimicking brain shallow and deep, machines learn when man is asleep, without complex equations or laws, making use of errors and flaws, regressive, divisive, and classifying, only in AI are so gratifying. They're gratifying because these are the terms used in AI: regressive, divisive, and classifying, regression techniques, and divide and conquer rules, and classification problems. Thank you. I stop here.
Thank you very much, Professor Ahmed. You have also given poetic and scientific touch to the presentation. If I may ask the question, since I'm very interested in DNA conformations, I just wanted to ask the MD that you have done for the genome analysis. The DNA is highly dynamic. It can form both canonical and non-canonical structures, non-Watson and Crick. So how do you take into account those secondary structures or conformations in your data? So
that's a very good question, and the answer is that the starting structure in each case is a bDNA structure. And from the bDNA structure, we basically let the system simulate under the standard Amber force field. We simulate it for 100 nanoseconds, and all the exhaustive tetramers have been simulated. So there are different tetramers. It may undergo canonical, non-canonical, or whatever the transformation it might undergo during the simulation process. And once you have the trajectory for, say, what we had in our data is about 10,000 snapshots over 100 nanosecond time, then each of these tetramers, we can say if this base is surrounded by these bases, 20% of the times it has this particular conformation, and 30% of the times it has this conformation. So this defines our conformational ensemble. And in the conformational ensemble, we do not use atomic coordinates. We use internal coordinates in the form of the shift, twist, these DNA parameters, which are called conformational or elastic parameters. So there are translational parameters compared to the ideal stacking, the base steps, how they stack one over the other, or how the base pairs with each other. So is there a stretching there? Is there a propeller twist there? So these are 12 conformational parameters which can be used to completely define a DNA structure, and an atomic structure can be reconstructed from that.
So what about looping and long distances? Sometimes there are large conformational changes. For example, enhancer coming in the proximity of the. So do you take into account those motions as well?
That's a bigger question. So because now the dictionaries are based on the tetramer simulation under the tetramer environment. So it is a 12 mers basically which was simulated. So this model can capture the local sequence dependent behavior. But if in order to, we have not done a looping modeling yet, but yeah, we are doing it currently. That is our interest also. But the looping behavior or the long-term folding of the genome will come across the accumulation of deformations, not from a single position. So for instance, a particular region is enriched in high shift tetranucleotides. So this shift will accumulate over a region, and this may lead to a looping. So these kind of questions can still be asked and are in the process.
Okay. Thank you very much. And I also would like to thank all the other speakers who kindly accepted our invitation at a short notice, and you shared their insights about how AI and ML tools, what are the opportunities, what are the challenges, and what are the fields from biology to chemistry to clinical perspective they can be applied for. And now over to Ravi for the concluding remarks.
I join Neeti. Very good evening. And I join Neeti in thanking all our speakers, right from the very initial, rather literary, erudite exposition, even without slides, by Dr. Narahari Shastri, to this poetic last grand finale from Dr. Ahmed.
Thank you.
It was very, very nice. But coming on a more serious note, this has been a very, very eye-opening webinar. And I think most students will agree that what we have grasped today is only a glimpse of the possibilities of AI, especially in drug discovery. We are yet to see large-scale applications in, say, toxicity prediction, although a little bit of hint was given by Dr. Subramanya, but using AI approaches and also trying to prevent early, we can intervene and lead to attrition of molecules which would otherwise go through a longer process at a very early stage itself. This would also be a wonderful application. So some of these things, again, work is at a preliminary stage, and I'm sure with more and more attention being drawn on all this, it will be wonderful. And I once again thank you all from the bottom of the heart of all my colleagues and students out here for wonderful lectures and presentations. Good evening once again.
Thank you.
Thank you all.
Thank you.
Thank you all.